Hmm. Okay.
Hello. Hello. Ali? Shall we start, sir?
Yeah, please start. One second, Anita. Sir, Vice Chancellor, sir, shall we start, sir?
Yeah, you can, sir.
Yeah. Anita, yeah, you can start. I think Pushpa Sarkar, madam. She is not logged in. Okay.
She was there.
She was there. Huh?
Yeah.
Okay, anyway, you can start. If the time is 10 o'clock, we can start.
A very warm and a very good morning to each and everyone who has logged in for a new mode of communication and meeting, that is a virtual one. It gives me immense pleasure to welcome each and everyone to the one-day National Virtual Seminar on Computer-Aided Drug Designing, a milestone in drug discovery. In chemistry language, it's called as CAD. It gives me immense pleasure to welcome all the dignitaries of DSU. I extend my hearty welcome to Honorable Vice Chancellor Dr. K.N. Balasubramania Murthy, Vice Chancellor DSU. I extend my hearty welcome to Dr. Pushpa Sarkar, Dean SAHS DSU. I extend my warm welcome to our principal, Dr. V. Murugan, COPS DSU, and to all the delegates who have logged in, including our honorable speakers of the day. Before going into the details, let me just give you an idea on what is in store for us the entire day. Well, our session has been divided into two parts, pre-lunch and post-lunch. Pre-lunch, we will have privilege of two speakers giving us brief introduction on their specialty, on their area of specialization in CAD, followed by a lunch session, followed by two more speakers on the various specialities on CAD. Then we have an open session for the feedback form as well as a question answer session. Let's not waste any more of our time and just directly go into the schedule. Before that, let's get a brief insight on the program. I invite Dr. Sonal Dubey, COPS DSU, to give a brief introduction on today's program. I hand over to Dr. Sonal Dubey.
Good morning, everybody. A very good morning to all the respected dignitaries, speakers, and delegates. Last 1.5 years, as we all have been witness to, has been a difficult and a different one globally. Most of the working places have shifted to home, schools and colleges have gone online, and so have many businesses. During all these unprecedented circumstances, we have seen how technology has come to our rescue. Today's webinar is to have an overview, another view on the technology, where we will see how technology, along with the molecular biology, chemistry, physics, and mathematics, has been put together to help us in designing new drug molecules. So, as today's webinar aptly titled as Computer-Aided Drug Designing, a Milestone in Drug Discovery. A rational approach to drug designing can be dated back to 1865, when Crum, Brown, and Fraser tried to relate biological activity of the molecules to its chemical constitution, that is, its structure. Science developed since then in this field, and the use of computers came into rational drug designing in 1964, along with a scientist named Hansch. With the advent of technology, both in terms of hardware and software, and our deeper understanding into mechanisms of drug receptor interactions, computer-aided drug design, along with being science, has become an art too. As most of our delegates attending the webinar today are students, I'm sure they are going to get an insight into what drug discovery is and how we can make use of computers to predict the structures of the molecules, possessing the desired descriptors, bonding interactions, and energies, so that ultimately we have a higher probability of getting a new drug molecule. Computer-aided drug design has come a long way, and its usefulness in terms of reducing the time and the cost in drug discovery is undebatable. Drugs like captopril, saquinavir, indinavir, dorzolamide are few of the examples showing the success of the role of computer-aided drug design in drug discovery. We have eminent speakers with us today who are going to highlight the various aspects of computer-aided drug design, like molecular docking, QSAR, etc. I'm sure you will have attainable takeaways from the deliberations and will be more intrigued by the field of computer-aided drug designing and would like to take it further. So without further delay, I welcome you all to this webinar and wish you an interesting and knowledgeable day. Thank you very much.
Thank you, Dr. Sonal. Now we have a brief idea on what is up in store for us. Let's begin the official ceremony by an address by our Honorable Vice Chancellor, Dr. K.N. Balasubramania Murthy, Vice Chancellor, DSU. Sir, over to you.
Good morning. It gives me immense pleasure to be present today morning on the inauguration of one-day National Virtual Seminar on Computer-Aided Drug Design, a milestone in drug discovery, organized by the College of Pharmacy under Dayanand Sagar University. I take this opportunity to thank Professor Murugan and his faculty team for extending an invite to be a part of the inaugural ceremony, and also take this opportunity to thank Madam Pushpa Sarkar for extending the invitation. This particular topic is very relevant in the context of drug design and rolling out the drugs to the market. When I look at the whole set of topics, the overall theme of the seminar is a computer-aided design. Computer is being used as a tool for designing a drug for a particular disease. If you try to look at the overall scenario as of now, approximately from molecular modeling to the releasing the drug or making the drug available in the market, it takes a huge time, long time of 8 to 10 years for things to get realized. With the aid of computer, I'm sure this time can be reduced a little bit, and the efforts are in that direction. I was just looking at the advantage that we derive by using computers in the drug design process. One of the advantages that we get is the speed with which we do the computations, trying to look at the efficacy of the whole thing, simulations, etc. Therefore, we are going to gain the speed. The second that we get by using computers is its accuracy. There's a lot of things that can be done. We can fine-tune it without much delay in time. It's possible to get accuracy. We can fine-tune the accuracy to the extent that we want. The third one is the cost. This can be done at a reduced cost because we do simulations before we go for the drug manufacturing and things out time. Thereby, the computer is going to give us the advantage of speed, accuracy, and the cost. However, there are several other advantages. Now, if you look at the overall scenario of drug design and drugs per se, drugs are normally being designed and rolled out to the market for curing a disease. However, there are multiple methods of the handling the disease. Of course, there's a preventive set of techniques, there's a curative kind of techniques, and prognostic things and diagnostic. If you look at the whole thing, I think there are several things which are involved in the process. Somewhere I have read that food that we take could be some form of drug if it's being used in the right way. It's possible to control many diseases by using the right kind of food, right kind of quantities, and the right combinations. Therefore, it starts with that. Then the second thing that I was trying to look at is to understand what exactly we mean by wellness of a human being or wellness of the scenario, wellness scenario. It appears mere absence of disease is not wellness, is not happiness. It requires 4 different components to be intact. The first component is physical wellness is one thing which we have to have. The second, the mental wellness. The third is the social wellness, how best we can live in a society. The social interactions is going to be critical. The last one is the recently WHO has added the spiritual wellness. Physical, mental, then social and spiritual wellness is defined as the happiness or the wellness quotient. If you look at the overall thing, the way in which it's moving, I'm sure going forward, a lot of changes are going to happen. And is also enormous amount of research that's going on in terms of the understanding the operations or the chemical reactions in a human body. It starts with the study that a human body is an interconnection of several subsystems, the digestive system, the nervous system, the several others. Now there is a good attempt that's been made to model each of the subsystems and also the interdependencies of the subsystems, how one subsystem is going to interact with the other subsystem. That modeling is going on. I think there's also a effort of analyzing the entire set of reactions in the form of model. There are also few attempts to build a mathematical model for a human interaction or human body. If that is going to happen, then the simulations become that much easier, that much well understood. Therefore, there's a set of efforts that are going on in modeling some of the subsystems. Probably if we can put all the subsystems together, we get a model for the human body itself. That's where one human being or one human body is different from the others. If you can understand that quite well, I'm sure the designing a drug for a given disease would become that much easier. At present, whatever the drug that we are using, more or less we use either a certain proportion of the drug for disease. For example, if I get the fever, I would go for taking a 650 dolo 650. And really, I don't know whether I need 650 milligrams or 650 dolo 650, or it can be cured with dolo 400 itself. We don't know, because right now we are going by some standard models without really understanding how much of drug is required for a particular human being. An attempt is being made in the recent past to personalize the medicine, personalize the drug. The amount of drug required by A may not be the kind of drug, the amount required by person B. Therefore, there are good research studies that are going on to do that personalized drug design possibly the future, it may happen or it may become a reality that a human being, all that has to do is to go to near a computer, get connected to the computer through a set of measuring devices. The computer would analyze and say, this is exactly what the problems with this human being. These are the kind of medicine that can be taken. That's the kind of research that's going on. That's the direction which is moving. Possibly in the future, the personalized drug design may become a reality. The other thing that's also being happening is to understand the entire function of the human body and to model it. The moment the model is known, then it becomes that much easier. In other words, it's all becoming a preventive kind of technology or a preventive kind of methods rather than the curative kind of methods. We can identify well in advance and ensure that the human body is, or the human being is away from any kind of diseases going forward, because the preventive care has already been taken. I think that's the best that can happen to the thing. I'm sure going forward, computer-aided techniques will become kind of reality in delivering the drug, both in terms of quantity and quality. I'm sure that's the direction which the entire process is moving. I'm extremely happy that the College of Pharmacy has taken a step in organizing a national seminar on computer-aided drug design. I'm sure the experts in the day would talk to you about the method that are used for various drug formulations. They'll also tell you how do they go about doing that. And I'm sure end of the day, you feel so happy for having attended this seminar and also get to know the latest trends in this area. I wish this seminar a grand success and thank all of you for the patient hearing, and thank Professor Murugan and his team, and also Madam Pushpa Sarkar for inviting me to be a part of this inaugural ceremony. I wish seminar a grand success. Thank you. Thank you very much.
Thank you, Dr. K.N. Balasubramaniamurthy, Vice Chancellor DSU, on a brief insight on today's program. I now welcome Dr. Pushpa Sarkar, Dean, SAHS, DSU, for a brief address on the gathering. Dr. Pushpa, Madam.
A very good morning to all the guest speakers, the participants, Honorable Vice Chancellor, Principal, the staff of the College of Pharmaceutical Sciences. Now, this particular seminar organized the computer-aided drug design, a milestone in drug discovery, which is the future of the drugs to be developed and discovered. This is the in thing to be discussed today. And I highly appreciate the team, Dr. Murugan and his entire team, for having organized this particular seminar today. And Dr. Sonal Dubey has given an insight of what it is, what is CAD, computer-aided drug design. And thanks to Honorable Vice Chancellor, he has elaborated and given a broad picture, the basis for why we require this. Now, moving on, you know that now with the enormous information available, and also the ease of communication, the world, you call it a global village as of now. Similarly, that every single field, technology has taken over. Any field, there's a technology. It might be the highest research, where you have all the elite research going on, you require technology. Maybe in the small tools, in the cleaning, also if you say about this, even there you have technology, the cleaning tools too. So that's the power of technology as of now. Now with this computers also have taken over enormously. It's just there everywhere. Coming to this computer-aided drug design, all of us know what is this computer-aided drug design? Now, discovery and development of a new drug is generally known as a very complex process. It takes a lot of time and resources. Now, take for example the COVID vaccine also. When we wanted this earlier, a vaccine to be discovered and developed, it would take years together. And now, because of the urgency and because of the facilities, resources, and the personnel available, they could discover this particular vaccine in a record time. I'm not taking this as a particular drug, but I'm just telling you how the technology has overtaken where a vaccine could be developed in a record time, thereby helping the entire world to come out of this particular pandemic, not entirely, but anyway, decreasing the morbidity. Now, as I was telling you, the discovery and development of any drug for that matter would require a lot of time. Now, this computer-aided drug design, it provides information about the chemistry and also chemical entities, which is basically inaccessible through laboratory experiments. So thereby reducing the cost and labor. Now it uses the computational approaches to discover, to develop, and to analyze drugs, and not only drugs, but also similar biologically active molecules. It gives valuable information about target molecules, lead compounds, screening, and optimization. The latest advancements, they provide a basis for the design of the ligands and inhibitors that require specificity. Now, all the pharmacists around here, you know the importance of this particular thing and what I'm talking about, the inhibitors, specificity of the inhibitors where they are required to block a particular enzyme, thereby providing, for example, even statins for that matter. All of you know what is the mechanism of action. So if at all there's a specificity in this particular inhibitors, which could be found in the development of this particular drug, it solves many, many diseases, particularly genetic disorders. Now, these computers will assist in the drug design effects, not only in predicting the binding mode and binding affinity of a molecule, but also how the drug will be absorbed, metabolized in the body, as well as its potential toxicity also. So the utility of computational chemistry is also for understanding of the three-dimensional aspects of the drug receptor interaction on a molecular basis and access the medicinal chemistry in designing of new therapeutic agents. Now, you know how it is, because unless earlier we had to just look into the two-dimensional structures or imagine the structures, or whenever you just extract a particular drug, it was just based on assumptions and things like that. But now you can just see the chemical, exact chemistry and all the groups in a three-dimensional structure. Once you see the three-dimensional structure, naturally, you know where the binding sites and whether it is the substrate or the inhibitors, how it goes and binds there, then how are you going to prevent the active groups in that particular thing? All of them, you can just, it's providing an enormous information for you, which you can specifically point out for the development. And in therapy, like, in treatment, This is going to really help a lot. Now, CAD approaches are all used very widely, as you know, to increase the efficiency of the drug discovery and the development course. By what I was telling now, by knowing the active groups, where they're going to bind. By such cases, you'll know the efficiency and how to increase the efficiency of this particular drug too, by modifying. Now, computational tools are widely used in pharmaceutical industries, in research areas, to improve the effectiveness and efficacy, both of the drug discovery as well as development of the drug. Now, CAD techniques are also used for rapid assessment of chemical libraries in order to guide and speed up. This is the most important part, is the speeding up. Speed up the early stage development of the new active compounds. Whenever you're discovering a new active compound, the speeding of the process. Earlier, it was just error and you just find it out with lab experiments, whether you're on the right track or not. Now, this is speeding up the early stage development over and above where you can develop and increase the specificity of that, or wherever you can introduce a particular group, modify the structure, etc. Now, the advance of CAD and the next generation whole genome sequencing, these devices, they provide opportunity for the discovery of new avenues of therapeutic approaches. Now, as of now, there are so many of these inborn errors of metabolism, genetic disorders. We know the cause. We know the cause for decades together, but we're not able to find the solution. Many times, only few of them we could find, but most of them are yet to be finalized. By this CAD, the computer-aided drug design, we can inch towards that. And many of these, because we have the whole genome sequencing done now. So for a particular disease, if you have a whole genome sequences, you can identify the disease. You can identify the gene which is causing the disease. And therefore the gene, hence the protein, hence you can have a target molecule. You can design a target molecule where it may help to target that particular the protein of the gene, where thereby overcoming this particular genetic disorders. Now, CAD will definitely improve the quality of research and facilitate the development of numerous drugs. Now, I would say that the CAD is offering much promise for the future of research, for the future of development of the drugs, for the future of discovery of new drugs too. Now, there are so many experts who would throw more light on these, the methodology and how it goes about, and extensively they're going to speak on that. So I wish all the best for all the participants here to imbibe, to listen to all these eminent speakers, and definitely to increase your, enhance your knowledge, and try to implement whatever knowledge you have gained today into the research and move on. And I thank Honorable Vice Chancellor for having given the opening speech here and elaborating the need of the health. And I also congratulate once again Dr. Murugan and the entire team for having organized this. Wishing all the best. Thank you.
Thank you very much, Dr. Pushpa Sarkar. Ma'am, it was a pleasure hearing you again. Now, I officially welcome Dr. V. Murugan, Principal, COPS DSU, to offer his address.
Good morning to everyone. Honorable Vice Chancellor, Dayanand Sagar University, Dr. K.N. Balasubramaniamurthy, respected Dean, Dr. Pushpa Sarkar, invited speakers, delegates, and faculty members. It's my immense pleasure to thank all the participant delegates for the one-day virtual seminar. Is going to participate and listen about computer-aided drug design, a milestone in drug discovery. Everybody knows that it is the hot topic in pandemic period. First and foremost, I heartfully thank to our Honorable Vice Chancellor, DSU, Dr. K.N. Balasubramaniamurthy, who accepted our invitation and had given an elaborate address on computer drug design, and also to address the gathering. Heartfelt thanks to Dr. Puspa Sarkar, Dean, School of Allied and Health Science, who has spoken very well about the need of computer drug design for the timing. I cordially invite all the speakers of today's virtual seminar who are looking forward for your valuable input on these topics. I welcome all the delegates and my colleagues for this virtual seminar. Welcome everyone on this day. Thank you.
Thank you, sir. For the kind note, the rescheduling of the sequence was done due to availability, non-availability of the speakers. Now, I hand over the program schedule to Mr. A.R. Mahesh Kops, DSU, for the introduction on the first speaker, Dr. Parashuraman P. Over to you, Mahesh, sir.
Thank you very much, ma'am, for your kind thing. So I would like to introduce our first speaker, Dr. Parashuraman P, who has obtained his bachelor degree in pharmacy, B.Pharm, and master's degree in pharmaceutical chemistry, M.Pharm, in Tamil Nadu, Dr. M.G.R. Medical University, India. As a UGC BSR meritorious fellowship, he obtained his PhD in pharmacy from Annamalai University, India. He has trained well in organic synthesis, separation, purification, and characterization of organic molecules using TLC and column chromatography, and interpretation by various spectroscopic techniques like FTIR, NMR, and mass spectra. He is well trained in drug design, lead optimization, QSAR, molecular modeling, ADME modeling, structure-based drug design, ligand-based drug design, homology modeling, pharmacophore analysis, analog design, DFT analysis, and molecular dynamics using computational tools. To this research credit, to his research credit, he has received several awards, filed and published 4 Indian patents, and published more than 45 papers in international peer-reviewed journals. I welcome you, sir, for this particular session, and I hand over the session to you to present your talk.
Thank you, Mahesh, sir. I welcome Dr. Parashuraman P. For the session 1, which is basics on CAD techniques and molecular docking. Over to you, sir. Dr. Parashuraman P.
Yes. Good morning, ma'am.
Thank you, sir.
Yeah. Can you hear everyone?
Parshuram, sir.
Sir, my voice is audible?
Yes, sir, your voice is audible. Your presentation is visible. You can go ahead.
Is it clear? Full view?
Yes, sir, it's full view. Full view is there.
Yeah. So I would like to thank you, Dr. Mahesh sir, and DSU people for giving me an opportunity and for a warm welcome. So, good morning, everyone. Today I'll be discussing the very basics of computer techniques and molecular docking. So people are speaking about drugs. And all. First, we should know what is a drug. So, drug itself, it may be either a chemical or biological, or either it may be synthetic or non-synthetic particle, which will be intended for the use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animal. This is said to be a drug. It may be either it's a chemical or biological substance. So we are saying the drug. From which sources we are getting? We are getting the drug from both the natural as well as the synthetic sources. Natural sources in the sense from animal, plant. Animals, insulin we are getting from pig and cow, from growth hormone, and from plants, digital is almost we know about the foxglove, and then morphine, those things. And then other than that, inorganic substance also we are using, and synthetic. Most probably around 80 to 90% of the drugs are from synthetic background only, either by chemical, biological, or biotechnological process. So we are speaking, many people are saying drug, and they are saying drug, again I'm synthesizing new drug, inventing new drug, every year inventing new drug. Example, if you take, we are studying the classification of the drugs. So in case of classification of the drugs, we are studying the same class of drug. We are synthesizing, or else we are inventing more and more of same class of drugs for the same disease. Why a drug is needed? Why? What is the need for a new drug? Every year we are finding a new drug that FDA have approved this drug, FDA approved this drug, that drug. So why? What is the need? First, we should know what is the need. So first need is unmet medical need. Unmet medical need. Because new diseases. Example, the most probably we are facing more than 1.5 years, the COVID, swine flu, etc. So in such cases, we need a new drug to treat such diseases and low efficacy. If you take a disease like a dementia or cancer, the efficacy of the drug is very poor. The efficacy of the drug is very poor, and side effects. If you take antidepressants or antipsychotics, not only those two, in case of other disease treatment also, it causes more amount of side effects. So we are not able to meet the requirement. So new drugs are required. And then the next one is cost of therapy. If you take the interleukins or Alzheimer's or spinal injury, for these diseases, the treatment cost is high, very high. It can't be affordable. So we have to find a drug which should be affordable by each and every person, each and every individual. Either it may be a poor economic country or high economic country, they should afford. And then to sustain industrial activity, that is the word we might have heard is patent. So till the patent is there with the company, then only they can able to fix their own prices and all. Once the patent expired, even the generic drugs manufacturer also will involve. So to overcome, to maintain such kind of financial commitments, everything, new drugs have been synthesized. And then the most important thing is drug resistance. So antibiotic resistance, anti-cancer resistance are those two which are more problematic one. So new drugs has to be synthesized, or new drugs has to be invented or discovered. So these are the reasons why we people involved in finding a new drugs. So drug discovery and development, the general process, what we'll be doing will be identifying the disease. So then after identifying disease, we have to isolate the protein involved in the disease. That is the pathogenesis, the process, the mechanism, how it gets involved. So for that, it will take 2 to 5 years. And then find a drug effective against disease protein. Towards that particular protein, only the particular protein, we have to find out a new drugs. So that will take 2 to 5 years. And then we'll be entering into preclinical testing. Then we'll be scaling up, or else we'll be making a proper formulation. Either it may be a oral or parental, whatever the formulation it should be required. Based on that, we'll be making. Then we'll be filing an IND, investigation of new drug. Before we enter into the clinical research, we have to file IND. We have to show all the documents, data of preclinical research works. Then only we can able to enter into clinical trials. So clinical trials, it will take 2 to 10 years. Then we'll be doing a NDA. We'll be applying for new drug approval. Then it will be approved, FDA approval. So these are the time taken for the process. Then, so these many things are in the process. Then what are the complexity? What are the troubles? What are the phases, different requirements we are facing? So if we take a 10,000 drug candidates for a particular disease, for a particular protein, with the valid biomedical hypothesis, the hypothesis should be very strong. Okay, this drug will work out for those things. Based on that, we'll be choosing 10,000 drugs. Then we have to filter out whether it is a targeted, whether it is selective, whether it is a potent, it's physically stable, soluble, permeable. It keep on going till manufacturable. So these all are the different steps which involve, and from 10,000 drugs to it will be found out as a one molecule, one drug molecule will be marketed. So this is how a drug process, a drug discovery process will take place. So nowadays we are speaking about the improvement in technology. So how technology is helping us? How technology is helping us in the process of drug discovery? So the general process is identifying disease and isolation of protein, finding a new drug. That is the common thing. So how technology is important? First one is by genomics and proteomics. So we have identified the disease, right, the protein. So based on that, these by applying the genomics and proteomics, we can able to find out the personalized targets, which is proper one, which is a prompt target. So that we can, if we are inhibiting that particular protein, the disease can be controlled, the disease can be cured. And then next one is high throughput screening. So we will be screening more than lakh compounds for a particular target. Then virtual high throughput screening. Both are similar, but this one high throughput screening is in vitro. We have to perform experiment. But virtual screening will be using through computer, predicting through computer. Then combinatorial chemistry, which is helpful in rapidly producing a vast number of compounds. And then molecular modeling, that is general what we are performing. Then in vitro and in silico admit models to find out whether the how is the absorption property, distribution property, metabolism and excretion. Then their toxicity can be predicted through this method. So these are the ways how technology is helping the people to overcome the overcome the timing process which will be required during our conventional method, conventional drug discovery process. So drug discovery technologies, and that first one is target identification, that is genomic, gene expression profiling, and proteomics will be involved. What I have explained there, I have made as a table. So then target validation, to find out whether the given target is proper or not, we will be doing gene knockout method or inhibition assay method. Then we have found out the proper targets, which is the protein, which are the culprit enzyme responsible, which are the culprit receptor responsible. We found out those. Then we'll be involving in the lead identification. We have to find the drug, right? First, we have to find out which lead will be suitable, which type of molecule will be suitable. Example, I have 10,000 molecules of different nature, different structures, different functional group structures. Like example, maybe the structural ring may have quinoline derivative, different derivatives of flavonoid derivative, pyridine derivative, like that. I have 10,000 molecules. So from that, out of that, which will be suitable, which type of molecule will be suitable, we will be finding out through lead identification. That is high throughput screening, fragment-based screening, combinatorial purpose. Then lead optimization. From that, after that, I have identified. Then I'll process proper way. From that, which will be suitable, what type of functional group should be present, whether it should be an electron donating group or acceptor group, how many number of donors should be present, how many number of acceptors should be present, like that I will be doing those things and all. So then preclinical phases. One is then we'll be, as usual, we'll be checking out the pharmacodynamic property, kinetic property, toxicity setting through animal. Then we'll be involved into the human trials. That is quite common. So technology will be helpful only till the preclinical phases only. It will reduces your time. It will reduces your cost in the till preclinical phases only. It won't help you in the clinical phase. So we have studied the word called CAD. CAD. So what is a CAD? Either it may be said to be a computational chemistry or computer-aided drug design. It is a chemistry whose major goals are to create efficient mathematical approximations and computer programs that calculate the properties of future drug molecules and thus helping the process of drug design and discovery. So here the computational chemistry is using the computer programs and mathematical equations. Using computer programs and mathematical equations, we'll be predicting whether this molecule will work out or not. Just like we will be saying, based on the properties, you will be saying a human. If you are saying how will be saying he is a human, he like by seeing the eye, hand, they are having two hands like that. And if you are classifying from a monkey, so monkey, what is the characteristic nature? So based on those nature, how we are saying, the same way we'll be creating a program. In such cases, this if it is having such kind of properties, this may work out, this may not work out. So that is the thing. Here we will be using mathematical calculation using computer programs. That is a CAD. And what is the need for CAD? Why we want to use CAD? So drug discovery today facing a serious challenge because of increased cost and enormous amount of time taken to discover a new drug. To discover a new drug, we are having too much of time, and we require more amount of money. So to avoid such kind of things and to avoid the competition between the different pharmaceutical companies. So the prime example, the live example, for past 1.5 years, we are seeing this for COVID, finding a drug for COVID. Many companies are in the tough fight and they found some vaccines. So that also can be done. So the time taken can be reduced and the cost which is involved in the drug discovery process also can be reduced. That is the reason why computer-aided drug design is required. So molecular docking. So molecular docking in general, what we can say is to find out, or in general, how a drug, how efficiently a drug binds to the receptor. This is just predicting how efficiently a drug binds to the receptor. That is at the molecular docking. In a simple word. So docking is the identification of low energy binding modes of a small molecule or ligand within the active site of a macromolecule or receptor whose structure is known. So this is just finding out the low energy binding, how it is getting binded, how a drug or how a chemical compound binds to the protein or a receptor. So in other term, what we can say is that it's a computational determination of binding affinity between molecules, that is protein structure and ligand. In other term, if a given protein and ligand, the binding free energy of the complex formed by docking them. So once it both come into contact, of the protein and ligand come into contact, they'll produce some energy. To predicting the binding free energy set at the molecular docking. So this is a ligand structure and a receptor, ligand receptor. In our biochemistry paper, we might have studied about the enzyme chapter. In enzyme chapter, lock and key model, Fisher model, right? For a particular lock, particular receptor only will open. The same way, so how it efficiently binds, how it efficiently opens the receptor, how efficiently it closes the process of a receptor. So we said a word called as modeling. We said a word called modeling. What is the requirement of modeling? So experimental determination of structure is still a time-consuming and expensive process. So in the drug discovery process, in the first slide, we saw identification of protein. Identification of protein. So for identifying a protein, it will take a time as well as it's an expensive process. That is the reason why modeling is required. One thing, and second thing is number of known sequences are more than number of known structures. So we have the sequence that is the gene sequence we have, DNA sequence we have. Those sequences are present. So based on those sequence, we doesn't have the proper structures. We don't have proper structures. That's why we can able to involve based on the sequences. And moreover, for a protein, the structural information is essential in understanding the function. How many beta sheets are there? How many alpha helix is there? So where is the twist and turns are there? How the protein is present? So based on the structure, we can able to predict what type of function it may have. So these are the reasons why modeling is required. The term in silico drug designing. So in view of pharmacological or else in general evaluation of a particular drug, what we'll be saying, we'll be doing the two methods in general, in vitro and in vivo. In vitro, outside the body, in vivo, inside the body, and both the terms will be typed or written in it should be described as in italic format only. It will be in italic format in any publications or anywhere. If you have seen, that will be in italic format. Here, if you are predicting the bioactivity, if you are predicting the activity of a particular compound using system, if you are predicting the bioactivity, biological activity of a particular compound using the system, that is computers, it will falls under the category of in silico. In silico. Why it is said to be in silico? Our systems, our laptops, our computers, what are the things? So these all are made up of silicon boards. These all are made up of silicon boards. That's why through silicon boards they are working on. So it is said to be in silico. So using in silico methodology, we are predicting the activity of a particular compound. We are predicting the activity of the particular compound. So this will fall under the term called as in silico. In silico is an expression used to mean that is performed on computer or via computer simulation. That's why it is in silico. You might have seen in your any research papers the term in silico modeling, in silico drug discovery process, like that they might have given. So types of in silico design. So what are the two in silico drug design? So there are two types of in silico drug designs are there. One is structure based, another one is ligand based. The term itself you can able to predict. One is structure based in the sense it is based on the protein structure. Next one is ligand based. Ligand based in the sense it is based on the ligand molecules. So that is the thing. Receptor based or structure based strategy. So uses this 3D structure of a target receptor to search for a potential compound that can modulate the target function. So I have identified a protein. I have identified a protein structure. Based on that protein structure, I'll predict the active site. Based on that, I'll predict the I'll predict the active site. Based on that active site, I'll try to find out a new drugs. I'll try to find out a new drugs. Example, Empro protein of a COVID. They have found out a new protein. So based on that protein, I can able to try to find a new drugs. So these involve molecular docking of each compound in the chemical database into the binding site of a target and predicting the electrostatic fit between them. So I have a protein structure. I have a protein structure, but I don't know which drug will go and bind. So what I can able to do, I can able to go to the drug databases. From the databases, I can able to screen for the particular protein, and from that, based on the best fit, I can able to choose which ligand can, which type of moiety, which type of derivative can be used. And then these compounds are ranked using as an appropriate scoring function. Scoring function and other things we'll be discussing in the upcoming slides. So such that the scores correlate with the binding affinity, how efficiently is binding. So receptor-based methods has been successfully applied in many targets. And then the next one is ligand-based strategy. Next one is ligand-based strategy. In the absence of structural information of the target, if I don't have a particular target, I don't know what is the proper structure. What is the proper structure for a particular disease, particular protein? I don't have any structure. Then what I can do, I can able to work out with the ligand-based method. Ligand-based method. So how in the sense example I will give, the drug ciprofloxacin we have been used. Ciprofloxacin and norfloxacin will have a similar structure and both will have a same sort of action. So it is based on the ligand. The drug discovery was based on the ligand structure. The drug discovery is based on the complete structural features only. So structures similar to known inhibitors are identified from the chemical databases by variety of methods. So I have a structure of a standard one, or else which have been marketed, which have been approved by FDA, or which is in clinical trials. I have the structure, so I can choose the structure and I can go for substructure search using PubChem database and many databases are there. I can able to feed that structure and I can able to find out the substructures which are relevant to that, which are nearby to that. So based on that, I can able to find out much more efficient, much more potent drug for the same class, for the same diseases. Some of these methods widely used as a similarity and substructure searching, then in pharmacophore matching and 3D shape matching. The pharmacophore modeling in the term, Dr. Manianda will be discussing in the next session. So he'll be much more he'll give clarification. So numerous successful applications of ligand-based methods have been reported. As I given an example, ciprofloxacin and norfloxacin. And then, so we have saying binding. It has to bind, it has to give the binding energy, binding score, something. What is the mechanism behind the binding? So complementaries between the ligand and binding site. That is the most important thing. How both get interacted. So stereocomplementaries, that is the shape of the ligand is mirrored in the shape of the binding site. Then only it will have a proper interaction. As I said, the example lock and key model. That is the most important complementaries. And second is physicochemical complementaries. So the basic binding mechanism, what we can able to say is, so how a ligand goes and fit into the active site of a particular protein. That is the thing. So this is the based on this only, it will give the score. Like chiral compounds we have studied, chirality, the mirror images. The same way this will work out. And then types of docking. So we have studied about in silico designing and those things. And that docking, in the two types of docking are there. One is rigid docking, another one is flexible docking. Rigid docking, what it will be, both will be rigid in nature. Both will be rigid in nature, and they can't able to move. However, we are keeping the same way it will be. Example, in a container to a container, if I'm throwing a stone, whatever the shape it is, it will go and sit as it only. That will fall under the class of rigid docking. In rigid docking, the internal geometry of both the receptor and ligand are treated as rigid. So, and if I put a stone, how it will go and sit? As usual, whatever the shape it is, the same way it will go. Next method is flexible docking and enumeration of these rotation. Either one of the molecules, either it can be rotated, performed. Every rotation of the energy is calculated. Later, the most optimum position selected. So, in flexible docking, how I can give example is the same way. If I'm so the same container, if I'm throwing some sand, so the sand, how it will be, sand or water, it will go and fit it. It can be adjusted. It can be adjusted. Here it will be adjustable. Here it will be adjustable in nature. So these are the two types of docking. And then categories. Type is different and category is different. So category, what are the two? Oh, because here for docking we need two things. One is the protein, another one is ligand. Rather than that, there is a term called as protein docking and protein ligand docking. So protein docking in the sense both inhibitor as well as the disease causing will be a protein in nature. Both are rigid. Both molecules are rigid. So here this interaction, if it both get interacted, what it will do? It won't produces any sort of changes in their conformation. Any changes in their conformation. And then the protein ligand docking. Protein ligand docking. So here ligand is flexible, but the receptor protein is rigid in nature. Ligand will be flexible, but the receptor protein will be rigid in nature. So here interaction produces conformational changes in the ligand. Conformational changes in the ligand. So here I have given the structure. So you can able to see A and B as such it will go on bind. As such it will go on bind and it won't produces any sort of activity. Sorry, any sort of changes. So computational modeling of quaternary structures only we have to take of the complexes found by two or more interacting biological macromolecules. Protein-protein complexes are the most commonly attempted targets for such modeling, followed by protein nucleic acid complexes. So here these two will be most probably used in the peptidomimetics peptide design. In such cases, protein-protein docking will be used, one thing, and in second thing, to create a new protein structure. To create a new protein structure, like if the proper particular structure is not there, for disease example, here the name is alpha 3 beta 2 helical structure. Let it be example. Then further the structure is that alpha 4 beta 2. I don't have the structure. What I can able to do, I can take this beta 2 part and alpha 4 part from the other thing, and I can able to club. Means I can able to do the docking, protein docking, to find out the proper structure of a disease. And then the next one is protein ligand docking. Protein ligand docking. So protein ligand docking is to predict the position and orientation of a ligand. To predict the position and orientation of the ligand when it is bound to a protein receptor or enzyme. Here I have given. So this cavity is said to be an active site. This cavity is said to be an active site. To that active site, how the drug got binded. How the drug got binded. You can able to see the drug structure. It is a 3D structure. It will get converted into 3D nature. So what are docking and scoring? What are docking and scoring? Docking in the sense to place a ligand into the binding site. So the drug structure is there. It can able to try to get into it will get fit. For a, I have bunch of keys. I have bunch of keys. Which key will go and fit? All looks like similar. So I'm trying 1, 2, 3, 4 like that. I'll be trying. So that is said to be a docking. Different pose. It will try to bind a ligand into the receptor in different poses. 1, 2, 3 like that. It can try to generate more things. And scoring is how for this one. What is the score? For this one, what is the score? For the third complex, what is the score? What for the process? To evaluate the ligand receptor interaction in a way that may discriminate the experimentally observed mode from others and estimate the binding affinity. Scoring will helpful in finding estimating the binding affinity of the particular compound. So components of docking. So if you take any software, it has to work with the two major components. One is search algorithm, another is scoring function. Search algorithm is nothing but generation of large number of poses of a molecule into the binding site. So this is the search algorithm. And then scoring function. This will calculate a score or binding affinity of a particular pose. So for this pose, what is the score? For this pose, what is the score? For this pose, what is the score? So that is the scoring function. The binding affinity or a score representing the strength or strength of binding. So scoring we said. So scoring we said. So this is the protein structure and the ligand got binded. So scoring function. So scoring function, on what basis it will work? On what basis it will be working? So this will calculate the basics, that is the hydrogen bond interaction between the ligand as well as the protein. their electrostatic potential, van der Waals force, hydrophobic interaction, pi-pi interaction, dipole-dipole interaction. So it will calculate a number of parameters. These parameters are based on the wave equation. This parameter is based on the wave equation. They have a proper calculation, means the equation. To that equation, these values will get substituted, and then the final output will be getting as a score. The score will be in negative. The score which we are getting is will be in negative. And many software, 99.9% of the software will give the docking score, binding score will be in negative value only. Like it depends. If you take a Autodoc software and Schrodinger, it will be -7, -8, -9, -10, like that it will be giving. So if a compound has given the score of -10, compound A -10 and compound B -7 and compound C -9, let it be. So here the compound which has given the -10, right, that will be the more potent, having a high binding affinity, So this is how they will be calculating. This is how they will be finding out which compound is best when compared with the test or other test molecules or the standard molecule. So they will calculate. In simple terms, it's just an interaction between the two friends. It's just an interaction between the two friends. If your friend is a neighbor, you may meet daily. The interaction will be different through telephonic call, through SMS, through WhatsApp, through Instagram. What are the options are there, right, for interacting? Here, the different interactions between the ligand and protein is said to be hydrogen bond interaction, electrostatic interaction, van der Waals force, hydrophobic interaction, and piper. So these physical terms will be keep on going. So we have discussed these things. What are the applications of molecular docking in modern drug discovery? How these will get helpful? So it determines the lowest free energy. It determines the lowest free energy structures for the receptor, ligand complexes, search database, and rank hits for the lead generation. Calculate differential binding of a ligand to two different macromolecular receptors. Study the geometry of particular complex. Propose modification of the lead molecules to optimize potency or the other properties. Library design, it will be helpful. Screening for the side effects that can be that is caused by the interaction with the proteins like proteases, cytochrome P450, and other can also be done. These are the application, the first application, and then second part of application is, so it is used to predict protein-protein interaction, and this knowledge will helpful in finding out the new pharmacological targets. This will helpful in the high throughput screening and less expensive, means virtual high throughput screening, less expensive than high throughput screening, and then this is faster than conventional screening, scanning a large number of potential drug-like molecules in very less time. So in my lab, if I have some 10,000 molecules, if I want to screen those molecules for a particular disease, I have to spend chemicals, I have to wait for, I have to spend time, everything will be there. The same 10,000 molecules, if I have in a structure, what I can do, I can able to do the docking study, and I can able to filter those 10,000 molecules into 100 molecules, with the top 100 molecules. From that 100 molecules, I can able to go further. High throughput screening itself a trial and error approach, means like 10,000 molecules if I'm doing in the lab, I don't know whether it will work out or not. So what I can able to do, I can able to predict those things and all. So in a very short manner, we can able to find out. So examples of some drug discoveries, means which have been obtained through computer drug discovery process. One is dorzolamide, sequinavir, relenza. So discovery of Indianavir, the HIV protease inhibitor, haloperidol as a lead compound in the non-peptide inhibitor for HIV. So these are the basics of docking. These are the basics of docking, what we have discussed. And then now we'll be discussing what are the prerequisites for carrying out molecular docking. So molecular docking, if you want to carry out a molecular docking studies, you should have a valid hypothesis. You should have a valid hypothesis. What is that valid hypothesis? A valid hypothesis, if example, in olden days, the traditional medicines have been used, but it is not into the market, scientific world. our grandmas, grandpas, our world and even in our home, our parents will be using some sort of natural remedy. For those natural remedy, we doesn't have a proper scientific evidence to prove that this is working on the so and so mechanism. We can use the molecular modeling means as a basic tool, the primary tool to prove those in a scientific way. What are the things? So plant A, let it be neem, because we know everyone neem. So this thing. Find out the ligand complexes, and if it is used for any okay, let it be bacterial infection. For bacterial infection, they are using. So how to find out? Now my hypothesis is clear. Already in olden days they are using this is for so and so things. So I just want to prove those things. This is my hypothesis. I have created a hypothesis. And then I need to optimize the protein structure. So what is the protein structure? Protein structure is a target. To which target I am trying to find the activity. Which target I am going to find the activity. So target protein I can able to download from protein data bank. Protein data bank. I given the next slide where the sources can be done. So example DNA, I have to find out what is the protein involved in particular disease, antimicrobial for most probably DNA gyrases response. Going to dog, I'm going to prove that neem is potent antimicrobial agent. That is my aim. So I found out this is the protein responsible. Next, ligands. So already the researchers have found and they have reported this plant is having so and so chemical constants, like around 250 compounds have been reported. So those structures either you can able to draw using ChemDraw, ChemSketch, Marvin Sketch. Many sketching tools are there for chemical compounds. You can able to sketch them one way. Second method is you can able to download from the databases, PubChem databases. You can able to type the compound name and you can able to download the proper structure of a particular compound. That is also possible. That is also possible. So I have found out which protein is responsible, what are the ligand complex. Then I have to go for molecular docking studies. So molecular docking softwares. What are the softwares available? First, we should know what are the softwares. So there are 3 or 4 ways we can able to say. One is web server. One is web server. Web server, how they will be working in the sense based on cloud computing methodology, they will be working. To website, if you have to go, you have to upload your protein structure, you have to upload your ligand structure. Then based on the protein and ligand structure, they will send the report to your mail. That is one way. Second way is freeware tools. Freeware, many tools which are available freeware. Freeware in the sense how they are giving many scientific institutes, scientific research institutes, they are trying to develop new softwares for the sake of finding new drugs. And that the most widely used software is Autodoc and Autodoc Venom. Autodoc is given the Scripus Institute. Scripus Institute, they are working on those things and they have given the software. That is also best tool. Autodoc and Autodoc Venom. Autodoc, we can able to do docking for one protein, one ligand at a time. We can't able to take 10,000 molecule or 1,000 molecule to do the docking in Autodoc. If you take Autodoc Vena, you can able to screen thousands and thousands of molecules to a particular protein at a time. In a single search, I can able to upload for doing those things and all. And academic freeware tools. Academic freeware tools in the sense, some software they may give trial versions or also some sort of beneficiary for teaching alone. CSAR is the software. CSAR is a software which they are giving for academic freeware. With that, you can able to teach to the students, but you can't able to publish your research data. For publishing research data, you have to get them on the commercial version. And then commercial tools. Commercial tools in the sense they have a license. Companies are working. Example, Scodinger, Discovery Studio is there, Gold is there. So those people will be working with the softwares. They will be they may have the yearly renewal or perpetual license are available. Using those tools, also you can able to carry out docking. These are the different methods of for carrying out the docking, and then visualizing tool. So once you have done your docking, you have to visualize what type of interaction, because we have studied many things. These are the ways how it is getting interacted. Hydrogen bond interaction, electrostatic, we have studied, right? So the same, how it got interacted, that image we have to see. That image we have to see. For those things, we require visualization tool, the complexes. Example, PyMol is the tool which we can able to see the interaction of a protein and ligand. One thing. Second thing is LigPlot is there, and third thing is in online web server PLIP, protein ligand interaction profiler. That's it's a web version where you can able to utilize those tools for finding or checking the interaction between a protein and ligand. What is the distance between them? Those things also can be measured through that. If you take the commercial tools, they have their own things. If you take Squad their master version, you can able to take the 2D and 3D interaction images there itself. 2D and 3D interaction you can take there itself, and then the Discover Studio also you can able to retrieve the image there itself. So these are the basic things. So once you found out what is the score from this report, you have found out those interaction. Then you might have done with the standard compound also. You can able to check where the standard compound went and binded with the protein that to the active site. Because the proteins are made up of amino acid, to which amino acid it went and bound. Next, test compounds. So to which amino acids it went and bound. What type of interaction it have created. So based on that, you can able to identify whether the test compounds are having binding efficacy as same of a standard or more potent towards the standard or less than the standard. So this is the way you can able to predict. You can able to predict whether the compound is having binding efficacy or not. So here are some databases. And then with this docking, one way I have given example. Second one is drug repurposing. For drug repurposing also, you can able to utilize the molecular docking molecular docking method. Why and how? Drug repurposing advantages is you can able to take the drugs which have been marketed by FDA, means approved by FDA. So in such cases, you doesn't want to carry out the toxicity study because those drugs are intended for human only. They have been used for human purpose only. So it won't create any toxicity. That is a major advantage. Second thing is why we can utilize what is a need to save the timing. It will be required to save the timing. It will be required. Example: aspirin drug was first it was identified as an NSAID. First it was identified as an NSAID. Then later they are the dose level they modified and they have given as an antiplatelet agent. The same way many drugs are available. So here why I said drug repurposing is many researchers they try to find out the drug for COVID through the drug repurposing process only. They have tried with antiviral drugs which are available, which are marketed. So they have chosen those drugs and they found they took the target of COVID and they have done the docking and they found out some sort of ideology. They found some breakthrough. From that, they have tried to find next. So it will be helpful in such a way. For lead identification, it will be helpful in that way. So other than that, what I can do is I can able to choose a database for a new disease or existing disease due to resistance, whatever the thing may be. With a valid hypothesis, I can able to go on download some databases. So in some cases, the few databases are IMPACT, curated for Indian medicinal plants, phytochemistry, and therapeutics. So here, from this, you can able to get the data structures of plant. Then AromaDB, Dr. Duke's, CMAP. So these are completely related towards the plant. This is related to the plant. That is phytochemical, which have been reported. Those things on that. It is there. So these are the database used. And then for chemical databases. One is zinc database, the most widely used one. They have more than 7 million compounds. Zinc 15 database, Chembl database is there. ChemSpider, Drug Bank, PubChem, TCM. This is for the Chinese medicines. And CHEBA, chemical entities for biological interest. So these are the different databases which are available for getting the ligands. If you want to explore more about the software, you can able to visit the website click2drugs.org. From those sources only I have kept here. So these are the agents which can be able to helpful which can be able to helpful for people to get the ligand structures. If I have found out some new protein, I have this protein, I have found out this disease. Example for malaria, okay, this disease is working. This for those diseases, there is no treatment, not proper treatment. You can choose the target. From the target, you can able to go to these websites. You can download these structures and do the docking studies. Then next one is protein. That is the target. Target database is most important. Most widely used is protein data bank. Protein data bank. So this will have protein, nucleic acid, and complex assemblies also. Some scientists, what they will do, they will upload the protein structure with the complex, with the ligand complex also, it will be uploaded. Then binding mode, mother of all databases. So this will also having a complete data history, PDB bind, NR list, that is nuclear receptor ligands and structures database. So here these are the databases, and most probably to the protein data bank, you can able to utilize the drug structures. Means use the protein structures can be utilized. This is how we can able to utilize. You can able to download your protein required protein. In case if your protein structure is not available, if your protein structure is not available in the database or anywhere, you have to carry out homology modeling. You have to carry out homology modeling. You have to take the sequence, then you have to do the homology modeling to find out the proper protein structure for the particular enzyme or particular receptor. That is the way. So we just came to the conclusion part. Molecular docking gives the promising effect on identification optimization in modern drug discovery. The combination of chemical information of a natural products with the docking-based virtual screening will play an important role in drug discovery in the post-genomic era, as more and more new potential targets are emerging from the functional genomic studies. So now the genomics have been genomics, proteomics, they have been growing fast. From those things, what scientists are doing for existing existing disease, they are finding the more potent target. More than the existing target, they are finding much more potent target. So if much more potent target is there, you have to find out the drug for those targets. So in such conditions, virtual screening will be more helpful. So docking based virtual screening lead to much higher hit rate than the traditional screening method. That is, rather than doing the high throughput screening, 10,000 chemical compounds I'm screening in the lab. So it will be cost and time. So rather than that, if I do the virtual screening, I can able to reduce those two, and much more efficient drug also I can able to identify. So docking method provides an opportunity for designing the active compounds. Whatever the thing may be, it has to be emphasized that docking based virtual screening is not the replacement of actual screening experiment. You have to be clear. The docking, based on the docking score, you should not expect that compound will be potent. It may be potent only out of think. Why? Because here in vitro study, some compound may be potent in silico. They may fail in vitro and in vivo. Some compound may be potent in silico. They may pass in vitro but fail in vivo. Some compound may be potent in silico, in vitro, and in vivo. Why? Because in vitro, it's based on the enzymes that may also pass. But in vivo, it has to cross n number of barriers. It has to cross the n number of barriers. It should be properly absorbed. It should be properly distributed to the particular site. It should be metabolized. It should be excreted. If it fails in any one cases, we can't able to blame the compound. So it is not the replacement. It will be helpful in reducing the time, reducing the cost of finding the medicine. That is it. As a matter of fact, these two methods are highly complementary. So this will support those methodology. Those methodology means for finding out the new drugs. So do you have any questions?
Ashram sir, there is one question over here.
Sir.
Like, which are the databases for polysaccharides? Which all tools can be used for docking of EPS with targets?
What is this EPS?
Srijish, I'll just unmute the participants so that they will be able to do. Srijish sir, you can unmute yourself and ask the question. Lipopolysaccharides.
Hello.
Hello, sir.
Yes, sir.
Sir, the question is direct, sir. Like, which are the databases that is available for exopolysaccharide, and even for docking studies, which software we can use with reference to the exopolysaccharide or the polysaccharides?
Exopolysaccharide, the molecular weight will be more, right, sir?
Yes, sir. Comparatively, yeah, not medium, yeah.
No, it will be more because it's a polysaccharide. You are saying the word itself a polysaccharide, right? So
Yeah.
you have to perform like based on the protein docking method. You have to go.
Okay.
Hex software is there. It's an online tool is available. Through that you can able to try that, sir. Hex.
Okay, sir.
I don't know about
Thank
the exopolysaccharide database. I'm not sure about because I'm working on with the ligands only most probably. So I don't have any idea about the exocellular polysaccharide database.
Yeah. Thank you, sir.
Thank you.
We have another question, Shankar Tapa. How homology modeling helps to find the protein structure?
Sir, if I'll take the recent example of COVID. They were starting, they couldn't able to find the drug structure. What they found the gene structure, I mean the sequence, the protein sequence. From that protein sequence, they have blaster sequence, PASA sequence will be there. From that, they blasted the sequence and then they found out the protein structure. So that protein, with that protein structure only, you can able to proceed for doing the docking studies. So homology modeling is the method of finding the protein, means like, generating the protein structure from the sequence.
Fine, sir. Thank you very much. There's another question from Vijay Lakshmi. The question goes like this: how to finalize the best template for homology modeling?
Ma'am, that based on the Ramachandran plot score. Ramachandran plot. If you are doing the Ramachandran plot, it will give the percentage where it is getting settled. So if it is 95%, 98%, 99%, it will be given in percentage. So based on that best percentage, you can able to find as which is the best one.
Thank you very much for answering the question, sir. If there is any other doubts, you can please raise it on the chat box so that we'll be clarifying it. Hello.
The session is open to all the participants for any questions. I guess we don't have any more of questions. So I take this opportunity to thank Dr. Parashuraman P for giving such a wonderful insight on molecular docking. Right, and now we move on to our next session. Before that, I request Dr. Prema Kumari KB to take over the session for introduction of speaker Dr. Manikanta Murahari. Over to Prema, ma'am.
Good morning, everyone. It gives me immense pleasure to introduce Dr. Manikanta Murahari. Dr. Manikanta Murahari is currently working as an assistant professor at Department of Pharmaceutical Chemistry, Faculty of Pharmacy, Ramaya University of Applied Sciences, Bangalore. His doctoral research involved design and synthesis of novel acridone-based derivatives and multi-drug resistance modifiers and their membrane perturbing potency in cancer cells using different computational modeling approaches at SVKMS Dr. Banubin Nanavati College of Pharmacy, NMIMS University Campus, Mumbai, as Junior Research Fellow funded by DOE, SERB. Post PhD, he joined as an assistant professor in Ramaya University and is actively involving in research activities of the department. He was involved in the process of designing and optimization of molecules from hit to lead and optimized lead for wide range of therapeutic targets. He is an alumnus of Manipal College of Pharmaceutical Sciences, Manipal University, where he did post-graduation studies in the discipline of pharmaceutical chemistry. His area of expertise includes structure and ligand-based drug design techniques, virtual screening, molecular dynamics, database searching, structure prediction, and analysis. He holds 25 publications in reputed international and peer-reviewed journals. Now, I welcome Dr. Manikanta sir to deliver the topic on applications of pharmacophore modeling in drug design. Thank you, sir.
Thank you, Prema ma'am, for a brief introduction on today's speaker. I now welcome Dr. Manikanta Murahari to take over this session on applications of pharmacophore modeling in drug designing. Sir, over to you.
I will be allowing him in a minute.
Yeah. So thank you, Prema ma'am, for that such a nice introduction. Yeah. So good morning, all of you. Hope I am audible to you guys. So
Yes, sir, you're audible.
let me share my screen. Yeah. So I hope you all could see my screen, right?
Yes, it is visible, sir.
Yeah. Okay. Thank you. Yeah. So today I'm going to talk on applications of pharmacophore modeling in drug design, right? So I'm just going to tell you one thing, like, so how this pharmacophore modeling help us in the drug design. So before that, I just want to make you understand that. So what is this pharmacophore, or so in what way this will help in design of the new drug molecules? So pharmacophore means generally, if you look at the basic definition, so what it says is, so what are the minimum structural features required to bind or to inhibit the certain targets, right? So what is the minimum structural features which is required? So this is what it is. How do we get to know what are the structural features are there? So for that, of course, we need to do some background study and all. Okay, so that we'll keep it aside. So now when you look at the drug molecules, right? So there are so many thousands of drug molecules are there. So if you look at these chemical structures of drug molecules, so what generally we see, like we see polar groups, non-polar groups will be there, hydrogen bond donors will be there, hydrogen bond acceptors will be there, or aromatic rings will be there. So any drug molecule you'll see, so there will be few features of this, or some drug molecules will have all these features, right? But still, we can tell that by looking at the structure, or of course we know about it. After seeing the structure, we can tell that this particular molecule is showing antibacterial activity. This molecule is showing anti-cancer. This molecule is having it will function on the, or it will work on the heart. This will be on CNS, right? So though every molecule will have the same chemical features, but they differ in the activity. So what makes the difference, right? So that is what we are going to find out by taking the advanced tools. So what is the difference is the position of individual feature within the molecule will make it different for different activity, right? That means, like example, if the position of hydrogen bond donor is at one place, so it will have this activity. If the position of hydrogen bond acceptor, the position of aromatic ring is there in this molecule, so that will have this activity, right? So that is how, like, to understand all this, so we need to know, I think you might have listened the previous session speaker. So all these drug discovery, it works on the concept of lock and key theory, right? So what they say is ultimately every lock will can be opened with one key. So we need to find that right key. So to find the right key, so for that, so you need to understand more about the structure of the lock. The better you understand the structure and design of the lock, the things becomes easier for you to select the right key. So if you, without understanding the lock, if you start searching for the keys, you may have to do more number of trials. So that is what a traditional drug discovery, what we are doing. So finding a right key, or the how much time you spend, how much money you spent on finding a right key for the lock, it always depends on how better you understand the lock, design of the lock, right? So that is what all these tools will help. Of course, what is the importance? So we are listening all these things from the morning. So all these traditional drug discovery, we are very familiar that it takes a lot of time, it involves a lot of money and all, and the process is very long. So it takes almost 10 to 15 years to launch a drug molecule into the market. So now as the technology is advanced, so these computational tools will help in reducing the timeline, reducing the time, money, and the manpower who are working. So even we have certain case studies where by taking the help of all these tools, what we are discussing today, so they could launch a molecule just within a span of 9 to 10 months. They could bring the molecule to at to clinical trials level. So that is the strength of this particular CAD. Okay, but so you need to understand. So it's like it is done by the machine, and machine doesn't have the brain. So one thing you have to remember, that machine doesn't have any brain. So human has to put the brain. So whatever data you give, you will get the results. So if you want to find the right key, you start exploring the design of the lock. The better you understand the design of the lock, the better key you could make it. Okay. So without much delay, we'll enter into the actual topic, that is pharmacophore. What is actually it is? So it is actually very enormously used application. Like this is the, when you start working on this drug design using these computational tools, it is always a better or a suggestion or an advice to start with pharmacophore modeling, right? So pharmacophore modeling, as I say, so you need to know what is the position of the feature for your target. So first thing is you need to identify on what target you're working. Like if you talk to researchers before some 5-10 years back, so they used to say that I am working on antibacterial, I am working on anti-cancer, I'm working on anti-malarial and all. But now we shouldn't say that, right? Because we have the advanced tools. So we should always tell, yeah, I am working on this target. So this is the target in, like, for example, you tell anti-inflammatory. So you should tell that you should not working on anti-inflammatory or something. I am designing molecules for COX-2. So all your research should start with the target, right? Because as I said, you're going to design a key for what? You're not you're not going to design a key for antibacterial or anti-inflammatory. You're going to design a key for the target. So your approach should be always, you should start with the target, right? So its application, so we'll see in further, like what is the actual process involved in it and all. But when you start your research work with the pharmacophore modeling, it actually complements your results, what you get from the docking or the end of the computational tools. So it will always be complement the results. So what does the definition? So he says that if you look at the split the words, these 4 rows. So 4 rows means it's a carrying, and pharmacophore means drug. So it actually tells about the features what a drug molecule should have. So pharmacophore, it is not a universal or something. It changes for target to target. As I said, the position of the feature in that molecule will makes it selective to target. So if the position of feature is in different place, so it will bind to different targets. Okay. So overall, it is nothing but the molecular framework carrying the crucial features accountable for a drug's biological activity. So what is the reason behind it, like, or how it makes the differences? So we can understand by looking at the first sentence. So when any drug molecule enters into human body, it will come across thousands of proteins, because inside the body there are number of proteins are there. So but still your molecule will not go and bind to the protein which it come across. It will go and bind only to the target for which you have designed. So it should happen like that only. So if it is binding to each and every protein which is coming across, so that will leads to many other side effects or toxicities. So all these side effects or toxicities, it is only because of the non-specific interaction. If you are designing a molecule with respect to one particular target, so definitely it will go and sit in that target only. So even though it goes inside any other protein which come across, so it cannot enter inside or it cannot sit there comfortably because you are not designed for that, right? The same way, if I give some other key and ask to open the lock, you cannot open it because that is not the right key for it. So you have to design a right key by looking at the structure of the lock. So without knowing that, if you start making it, so then it may open other locks. So that should not happen. So all these things are possible only when you know about the size and shape of the target or the size and shape of the active pocket. So like every molecule, a molecule can go and sit inside the active pocket, but it cannot block the function of that until unless the molecule go and sit inside the active pocket and it has to form certain interactions. So right size, correct shape, and complementary chemical features. So sorry that yeah, so right size, correct shape, and complementary chemical features are the key factors for protein drug recognition. That means you need to understand more about the size of the active pocket, shape of the active pocket, how well it is, and what kind of chemical features are there in the active pocket. So like for example, you need to know what is the chemical nature of the active pocket. So what is this active pocket? Active pocket is something so where it is a sequence where any substrate or any molecule comes to it. So whatever amino acids are there in that active pocket, they only will respond to it. So entire protein will not respond, right? So that we know about it. So active pocket plays a major role. So for that, you need to understand what is the sequence of the active pocket, what kind of amino acids are there. So amino acids means we know the chemical classification, right? When we have studied the classification of amino acids in terms of chemical features, like positively charged amino acid, negatively charged, polar amino acid, non-polar amino acid. So these kind of chemical nature will help you in designing your molecule, right? If there are more number of polar amino acids are there in the active pocket, so then you may have to put more polar groups in your molecule. If there are more non-polar amino acids, you have to put non-polar groups in your molecule. Otherwise, if your active pocket is polar and if you try to put non-polar groups in your molecule, it cannot go and sit there, right? So you need to understand. So whether hydrogen bond donors are there in your active pocket, put hydrogen bond acceptor in your molecule so that they both will react, they'll form a hydrogen bond and it will get stabilized inside the complex and it will try to block the functions of that particular enzyme. So you should always understand the size and shape. So both these corresponds to all these, what is the size of the active pocket accordingly. If the size of the active pocket is small and if you try to prepare a bulky molecule, it cannot go and sit inside. It cannot enter into the active pocket or it cannot sit inside it, right? And complementary chemical features. So this is also important. If positively charged groups are there, you put negatively charged in your molecule. If negatively charged are there, you put positively charged in your molecule. So it is a better and fast tool to recognize a molecular similarities. When you compare with other techniques like docking or QSR or dynamics, like whatever computational tools, if you compare, so pharmacomodeling, so it is one of the better tool and it will, all these computational calculations also, it doesn't take much time. So it will work very fast and you don't require much computational power. You can just do it in your laptop itself. And the time it takes also, it will be in the seconds only. For each molecule, it will take hardly in a fraction of seconds, right? So what are the general pharmacophore features? As I said, so these are the general features which you find in every molecule. So some molecule may have 3-4 features, some molecule will have 2-3, or some may have 5-6 features also, some may have multiple. But only the difference is the position of this feature in the molecule will make it different for different activities. So now all our effort is to find this only. So if you're designing a molecule for one target like COX-2, so in the COX-2, you need to know what is the position of hydrogen bond donor or what is the position of hydrogen bond acceptor. So for that, you need to know what is the position of these individual features in that. So that will be complement to the active pocket, right? So I'll just take this simple case study about COX-2 inhibitors. So we know these COX inhibitors are very familiar and like we take it regularly. right? So cyclooxygenase. So it is mainly responsible for inhibition of all these prostaglandins and all, right? So initially they were like we were using non-selective COX inhibitors and all. So but later mechanistic studies done by the scientists. So what they've identified is, so in our body, so there are two types of enzymes are there, COX-1, COX-2 are there, and the functions are different. So the prostaglandins which are produced by COX-1, these are good prostaglandins, and the prostaglandins which are produced by COX-2, these are bad. So they are nothing but inflammatory mediators and all. But so far, without knowing the structure of COX-1 and COX-2, so we started designing molecules and we are using it. So that's why now we are experiencing the side effects and all, right? So now we know whenever we go to a doctor, so by default, whenever there is any painkillers, analgesics are there, so default they are giving antacid, right? Antacid or anti-ulcer drugs or something. So first of all, we are suffering from some one pain. So on to that, if you give another molecule, so another drug molecule, that means you are adding the side effects for that, because we know that none of the drugs are free from side effects. So every drug will have a side effect, and if you are using it for long term, so we may end up with some other things, right? So for that, what is the solution is? So you have to understand more about this COX-2 enzyme, and you design a better inhibitor. So you have the lock now, so you design a better key. So then they started exploring the structure. So what is the structure difference between COX-1 and COX-2? So by looking at this, we can make out. So COX-1, the active pocket is very small, and COX-2, the active pocket, you can see the shape of the active pocket, it is bigger. So that means, so whatever COX-1 inhibitors they have synthesized, they are small molecules and all. So that's why they could easily enter into both COX-1 and COX-2, and they are showing the activity. So they are blocking that. That's why we are relieving from the pain or fever or something, right? So studies are going on in that process to design a better COX-2 inhibitor. So in that process, they've identified, this is the review published in Nature in 2003. So what they say is, so whatever molecule you're designing, that should go and bind to these 3 amino acids. If you're able to design a molecule which can bind to these 3 amino acids, and if it fits in this shape, so definitely the chances are very less for that the same molecule to go and sit in the COX-1 pocket, right? So that's why if you're able to design in that way, so that will help in identifying a safe molecule, so which is free from other side effects. So we can use the molecular docking technique, what was discussed before, and we can get the databases, chemical databases, and screen them and find which are the molecules which can bind to these amino acids, so that it will not have any chance to go and block the COX-1, right? So this is the basic understanding. So that means what you need to know is, so when you want to do the pharmacophore modeling, so you need to understand more about the structure of the target protein. So you need to have a target protein. So the target protein, so either you can get it from the PDB, X-ray crystallography structures are there, plenty are there. If you are not getting, so then as discussed, so you can use the technique called homology modeling. So once you have the sequence, so you can find the structure of the protein, and then so there are tools to predict the active pocket. So you can predict the active pocket and try to understand, so what is the chemical nature of that particular active pocket. So the things will become easier only when you have the structure of the protein, right? So for that, you need to focus more on the 3-dimensional structure of the 3D structure of the protein. 2D is not sufficient. So you need to understand the size, the shape, and the chemical features. So these plays a major role in designing the molecule. So once you have that, then you can easily design it. So we know the beauty of the proteins. So though all the proteins are the sequence, if you look, so it's only 20 amino acids, but none of the 2 proteins will have identical sequence. So that is another beauty of the protein. And these proteins are also designed in such a way that like proteins are nothing but they are tools for our body, right? So they are nothing but the tools. So you need to understand, so what is the size and shape of your protein? So that only will help you in designing a better medicines. So design of lock helps in making the key. So like how we know, as I said, these proteins are nothing but the tools which are there inside our body. So by looking at this tool, we can tell that what is the purpose of this. So what is the purpose of this funnel? What is the purpose of screwdriver? And what is this eggs rack and all? So because these are designed in such a way to use that for that purpose. So what this tool can do, this cannot do, and what this tool can do, this one cannot do, right? So the same way in our body also, all these proteins they are designed to do that kind of function. The proteins are everywhere. The proteins are there in the body, but the proteins, the functions, what is done by the protein which is present in hair and nail are different from the blood, muscle, cellular messengers, antibodies, construction workers, enzymes, brain and nerves. So everywhere proteins are there, but all those proteins, the size and shape of the protein will be in such a way that what function it has to do. So based on that only it is designed. So that's why. So what I mean to say is active pockets, as how the sequence of proteins are different, in the similar way, the sequence of active pocket is also different. The size and shape of the active pocket is different. The chemical nature of the active pocket is different. Because the size and shape of the protein or the chemical nature of the protein is designed in such a way so that it can do that function properly. So you need to understand more about it. So we have the techniques called X-ray crystallography and all. So by that, we can find out the structure of the protein. So once you have the structure of the protein, so then we can design a pharmacophore for that. So like you may get it out, like, so whenever any new molecule, when a new disease or new disorder has come, something like this COVID, so we don't have any protein structures. So then how do we, or we don't have the sequence, at least if we have the sequence, so you can build, you can use the homology modeling tool and you can find out, you can generate a pharmacophore modeling, right? So, but still, even if you don't have also, so there are different ways to generate a pharmacophore modeling for a particular target. So these are the different ways, some of them. So from diverse active ligands. So if you have any molecules reported for that particular target with the activity values, okay, from active and inactive ligands in a congeneric series. So I said there are two differences between diverse active ligand and congeneric series. So what is the difference between them? Diverse active ligand in the sense, so there is no, I mean, the structural features are different. Like for example, so diverse active ligands means there will be different type of heterocyclic rings will be there. So one class quinolines are there, then quinazolinones are there, indole are there, benzimidazole are there. So all are having different moieties. So that we call it as diverse active. So active and inactive ligands in a congeneric series. So congeneric series means you have a series of quinoline analogs, for example. The only difference between first and second, third molecule is the position of the groups. So quinoline means, so onto the quinoline, in the first molecule, the position of chlorine is at 1. The second molecule, 2. So in the third one, it will have a methyl group. In the fourth one, it will have other ethyl group or something. The same series, but with slight modifications in them. So if you have that also, you can generate a pharmacophore model, but you have a diverse ligands like indole, benzimidazole, quinoline, naphthalene or something. So if you have different heterocyclic rings, then also you can generate a pharmacophore model. A protein ligand complex. So if you have a protein ligand complex, that means you have a crystal structure of the protein with its inhibitor sitting in the active pocket, then also you can generate. If you have only 3D structure of the protein, then also you can generate. Still, you don't have anything, but you have only a potent molecule structure. So then also you can generate a pharmacophore for this. So there are different ways are there. You don't have to limit your research. So there are different ways are there. So by trying all the different ways, all the possible ways, you can generate multiple pharmacophore models, and you can go ahead with your research. So we'll just see for two cases how we can go ahead for generating a pharmacophore model. So first thing is we'll discuss about the ligand-based pharmacophore model generation. So what is this ligand-based pharmacophore model generation? The title itself tells you that it is ligand-based. That means you don't have a crystal structure of a protein. You don't have a 3D structure of the protein, but you have a set of molecules which are designed, or you have a set of molecules with their experimental values. That means IC50 values you have. So it can be of diverse or it can be of same congeneric series. So you will go for this method when you don't have a crystal 3D structure of your target protein. So what is actually, how does it do is, or what is the criteria required for it is, the thing is you need to have a set of molecules, at least you should have a good number. The criteria is you should have a good number of molecule. Good number in the sense maybe somewhere around 50 to 60 molecule. So that is the first criteria. Okay, so it will have a different criteria. So first criteria is you need to have a good number of molecules, at least 50 to 60, that is one. And the second criteria is you need to have the activity values should be of broader range in the sense it should have active molecules, potent molecule, moderately active, and then inactive molecule. So inactive means if you can understand the units of IC50, so we report it as micromolar or nanomolar range, right? So when you talk about that in that way, for example, you should have a molecule which have an IC50 of 100 micromolar or more than 100 micromolar some molecules, 100 micromolar some molecule, 50 to 100 micromolar some molecule, 1 to 50 micromolar, 1 to 10 micromolar, and again in nanomolar range, you need to have some molecules. So that means you need to have molecules in different ranges. Okay. So when you have a molecules in different ranges, so all those molecules structures with the IC50 values, if you give it to the software, you just have to collect those molecules, draw those molecules in the software, and you have to give it to the software. So what does it do is, so it will try to collect, let us say we have collected 60 molecules with different IC50 values. So what does it do is when you give it to software, so it will see what is the first molecule. So in first molecule, how many pharmacophoric features are there? So let us say it has hydrogen bond donor, hydrogen bond acceptor, and hydrophobic groups. So these are the 3 pharmacophoric features which are present in all the 60 molecule. Just as a case, as an example, I am telling. Okay. So in the first molecule, what is the position of hydrogen bond donor, acceptor, and hydrophobic group? What is its IC50 value? In the second molecule, what is the position of individual feature? What is its IC50 value? So in the third molecule, what is the position? What is its IC50 value? So in that way, the system, the software will analyze what is the individual position of the features and what is its IC50 value. So it will map each and every molecule's positions, position of features of each and every molecule, and it will correlate with the IC50 value, and it will tell you what should be the position of individual feature if you want to design a better molecule. Let us say if you want to design a molecule of 1 nanomolar activity, where the position, where should be the hydrogen bond donor position in a molecule, where should be the hydrogen bond acceptor position in a molecule, where should be the hydrophobic group position in a molecule. So that is what your software is going to tell you. So now when you have both active and inactive, so inactive molecules will tell you, that will help you in giving a model where should not be the position of hydrogen bond donor and where should not be the position of your features. So that is why I'm telling you should have molecules of different ranges. You should have active, moderately active, and inactive, so that whatever model you are getting later stage, so that will help you in picking a right molecule. So for example, if there is no much difference in your activities, so then the model, the pharmacophore model, whatever is coming, that cannot tell you whether your molecule will be active or not. Okay, so this is the criteria for which you need to identify. So for that, you need to identify, you need to do the literature and find out what are the, you should have a good data set. You should collect 50 to 60 molecule with different IC50 values, right? So for example, this is one of my research. So where I have generated a pharmacophore model for COX-2 inhibitor. So this is how will look you. So it looks like. So it has a 4 features. So acceptor. So if you look at that feature AHHR, that means 1 acceptor, 2 hydrophobic groups, and 1 aromatic ring. So these are required for your design of COX-2 inhibitor. And it will also tell you where should be the position of individual feature and what should be the distance between individual feature. So if you generate a ligand-based pharmacophore model, so it looks like this, right? So this should be the feature. If you have the features in this position, so then it will have a chances to inhibit the COX-2 enzyme. Okay. So as I said, what should be the process? The process is, so you should always pick the right set of compounds with their chemical structures, right? And so another one is the conformational search. So like conformational search means what I say, every molecule will have infinite number of conformations. So when I say that these are the 4 features required, now if you turn the molecule in different size or different shape, then sometimes every feature may match, right? So for that, you need to know, that means you need to generate a multiple conformers for every molecule. So it's not like how you write it on a paper, the structure, it will not exist in that, right? So there will be infinite number of conformations will be always possible. So your software, that the software will take care of it. So it will, for every molecule, it will generate multiple conformers and it will consider each and every conformer. Okay. And then, so feature extraction and representation. So as I said, though you take 60 molecules, so 60 molecules, when you give it to software, so it will generate multiple conformers. So like some softwares have an option. You can generate for each molecule, you can generate 50 conformers. For each molecule, you can generate 100 conformers. That means for each molecule, let us say if you tell the software to generate 100 conformers, that means the 60 molecule will be into 100, 60 into 100, that will become 6,000 molecule. So 6,000 molecules, it will overlay on each other and it will locate the position of individual feature and it will see what is the activity value. So conformational search is also one of the very important parameter. So it will generate conformers, then feature extraction and representation. So feature extraction means it will locate what is the position of individual feature in every conformer of every molecule and it will correlate with the IC50 value. So once all that is done, then it will identify the pattern. So what should be the pattern of, or what should be the pattern of your features, or what should be the pattern of your pharmacophore model. So and scoring. So some softwares will give you multiple pharmacophore models with some scores. Okay. So once you get all these scores, once you get some 5-6 models, so then what you have to do is validation. Validation is also very, very important. So before going to use this model for filtering chemical databases, you need to validate your model. So that means, so for example, once you have a 60 compounds, so you have to separate some 10 compounds for as a validation set. That means all the 10 compounds also should have all ranges of molecules. That means you should have active molecules, inactive molecules, moderately active molecules. Validation is also very important. So once you have these 4-5 models, so then you take your validation set and you screen all these 5 models and you see so which model is picking a right molecule, right? So then it will give some score for every, once you pass the database through that, or once you pass the validation set, so it will give some pharmacophore score. Score means that score will tell you how well or whether the molecule has the required features and whether the molecules has the required features in the required positions or not. So that also, like how we see docking score, in the same way we get the pharmacophore score also, you will get it, right? So molecular alignment, handling ligand flexibility, and proper selection of training set compounds are considered as biggest challenges. So sometimes you getting a right data set is also very important because you should have a wide range of activities. So as I said, so it should be more than 100 micromolar, less than 100 micromolar, less than 50, less than 10, then 1 nanomolar. So getting this set of compounds for this target is a challenge. Okay. So if you get those molecules, so then you can give it to the software and you can generate a pharmacophore model using that. Okay. So we have a short video on this ligand based. You can just watch it. So I think it is a bit more detailed. So what I have told is just I have given an outline. So there are different features, are there different options are there for you to play with different softwares. So the video was for the Schrodinger software. So Schrodinger software you have, of course, it's a commercial version. So you have multiple options are there where you can play with. So how many features you want? Like for example, from the literature search, if you know that, so your hypothesis or the pharmacophore model, what is it is generating, it should have at least one aromatic ring. So you can fix that. So it requires one hydrophobic group or one aromatic ring. So you can play. There are multiple options are there. But what is the requirement is that so you need to have a good number of molecules with different activity ranges so that it will help you in generating a better model. So the accuracy of the model or the prediction level of the model always depends on or how your training set is there. So what type of molecules you have taken and what is their IC50 ranges. So if you have a better, better ranges are there, then you can get a better model, right? So it's always depends on the type of molecules and their IC50 values. So next, another case study, what I'm going to discuss is structure-based pharmacophore model generation. Okay, so structure-based, that means when you have a crystal structure, when you have a target structure protein, so how you can generate that model, how you can generate a pharmacophore model. Okay, so either you have a 3D structure of the protein alone or receptor ligand complex. So sometimes you will get receptor ligand complex also you will get it, right? So in both the cases, so you have to get the 3D structure of a protein or the 3D structure of the complex and you have to give it to the software. So once you give it to the software, so what does it do is the software will first read what is the sequence of the active pocket. So what kind of amino acids are there in the active pocket? What is their chemical nature? So based on that, it will read the active pocket and it will tell you. So as I said, there should be always a complementarity between your molecule and the target protein. Then only they can comfortably sit inside. So first objective is your molecule should go and sit inside the active pocket. So for sitting inside the active pocket, there should be complementarity. Complementarity means polar groups are there in the active pocket, means your molecule also should have a polar group. So then polar, they can have a different kind of attraction and they sit. So if polar, non-polar is there, then there will be a repulsion. So similarly, if positively charged groups are there in the active pocket, your molecule should have a negatively charged. So in that way, so it will try to read the size and shape of the active pocket, the chemical features of the active pocket, and it will tell you what should be the pharmacophore model for this particular target. So by chance, if you have a complex, so when you have a complex means, so it will have it can be a drug molecule or anything which can be an inhibitor. So inhibitor, like I always suggest, whenever you pick a structure, 3D structure of your target protein from PDB, let us say you have a cyclooxygenase 2, you want to design for that particular target. When you go to PDB, if you type cyclooxygenase 2 in your search box, you will get multiple number of 3D structures. So you may get it out which one I should take. So I suggest you should always go for the PDB ID where you have a ligand for that particular structure, and preferably that ligand is a drug molecule in the market. So if you are not getting a drug molecule, you can just see what is the ligand which is having IC50 value, or the ligand should be a potent inhibitor. At least it should be in a market or in the clinical trials. So if it is not even in the clinical trial or if it is in preclinical, so I always advise that. So you should always pick a PDB ID where the molecule is in at least in preclinical stage or clinical stage or something, or at least some experimental work is done and it is reported as a potent inhibitor for that target. Okay, so in that way you have to pick. So once you pick that, if you give the complex to the software, so what does it do? So anyhow, the molecule which is there, so you have a bioactive conformation of that particular molecule. So try to understand bioactive conformation. What is the bioactive conformation? That means that is biologically active conformation. So that is the conformation what is coming is nothing but biologically active. And that molecule is already reported as a potent drug. So that means whatever molecule you are designing, that molecule also, if it sits in that position and if it is forming interaction with the similar amino acids, So then your molecule also, you can expect that, so that molecule also will be having closer to that. So it will also may have a better activity. So let us say that one drug molecule is sitting in the active pocket and it is forming 2 or 3 interaction. Now, whatever molecule you have designed, it is also forming 2 or 3 interaction. And if it is forming an additional interaction and if it is stabilized more, so then that molecule may have a better potency and all, right? So in that way, so what does it do is, once you have a ligand in the active pocket, it will read the, so what is the complementarity between the ligand and protein, and it will give you the feature, what should be the position of the features. So if you look at the process done by the software is, so once you give the complex, it will identify the active site. So what is the active pocket, and it will construct a complementary image. So it will construct a complementary image, construct a complementary image in the sense, so what features are there in the ligand, what chemical features, and what chemical features are there in the active pocket, whether both are complementing each other or not. If they are complementing, which features are complementing. So as it is experimentally proven, so then whatever features which are complementing, so that features your unknown molecule also should have. So then it will generation of queries, searching and hit analysis. So it will generate multiple images and it will give you, so one particular position, like one particular model where it will tell you this should be the position of individual feature. So when you want to design a molecule for this particular target. Sorry. So in that way, so if you give a complex to the software, it will analyze the active pocket. If you give it with the complex with the ligand, it will analyze the features of ligand, it will analyze the features of active pocket, and it will see which are complementing. And based on that, a series of images it will construct, and it will give you, this should be the pharmacophore model for this particular target. So in that way, it will generate and it will give it to you. So for the same thing, there's one more video for structure based. You just watch it. Yeah, so that is of structure based. So it is also same of Schrodinger software. So you have a different options like as it is shown, auto, pharmacophore, and manual. So where you can do is like manually you can pick what features you want, or you can allow the software only to pick what all features. But if you allow the software, so it will try to see it will catch only the complementary features between the ligand and the protein, and it will give you, right? So in that way, there are multiple softwares are there. You can take help of those multiple softwares and you can generate a pharmacophore model either by structure based or by the ligand based, and you can use them for the screening of large databases, right? So once you screen the large databases, what will happen is, so like I was telling, you should always begin your research with pharmacophore modeling. So why it is so when if you start with the docking, what will happen? The one thing is when you have a chemical database, if you start like let us say you have some 10,000 molecules. So if you start right away 10,000 molecules and give it for docking, so it takes more time and you need more computational power also, right? So in the other way, if you start with pharmacophore modeling, so what is this pharmacophore modeling will do? So in that 10,000 molecules, so it will see out of these 10,000, which molecules are having the similar features which what I require. So for you have the model and you pass all these 10,000 molecules to the model. So once you pass it, this model will pick you the molecules which are having features what is matching or what you require. So then that 10,000 you can cut down the 10,000 to maybe 2,000 or 3,000. So once you take those 2,000 or 3,000 molecule, and then so if you do the docking, so you can save a lot of time, because pharmacophore modeling, it won't take much time because there is nothing. It just has to see whether like once you give it to software, so it will pass the molecule to that through that pharmacophore model, and it will see whether it has the features or not, then whether the features are in the right place or not. So only this part it will see. It won't take much time. But when it comes to docking, it is different. It has to go inside the active pocket. It has to sit in the active pocket. Then it has to make an interaction. Then it will calculate the energy of that interaction, whether it is stable or not. Then what residues it is doing. So all it has to see so many parameters. So the time it takes will be more for docking. So you can save the time and you can reduce the computational power. You don't require much computational power, and you're refining the database. You're picking the database which you are doing only for the molecules which have required features. So there is no point in checking in every molecule, right? So you know what features are required for your molecule. Whether the molecules have the required features, then only you do the docking and check the binding affinity. So if your molecules are not having the required features, then there is no point in calculating the binding affinity, right? So if you have the required features, then you calculate the binding affinity. So in that way, that will serve that logic. Okay. So what are the different applications? I have just listed some 2-3 applications. So virtual screening. So virtual screening is something like you have a large number of chemical databases are there. You can just screen them. You generate a pharmacophore model, and so it is the answer like what I just explained. So you can find screen the database and find the molecules which are having the right features. So that will save a lot of time and it will serve the logic also, right? So usually it will be applied implemented at the beginning of the process. So and we have a large number of libraries are there. So just now Dr. Parshuraman, he has told, so what are the different libraries available and all. So if you look at the workflow, so once you generate the model, so you can generate a ligand-based model or a structure-based model and screen the databases and do the molecular docking. And then once the docking is done, so from there you will get the good compounds which have a good binding affinity. So once you get the list of compounds which are having good binding affinity, then you check, excuse me, you check whether they pass this ADMET filters. That means pharmacokinetic, absorption, distribution, metabolism, excretion, and toxicity. So if they pass that, so then you go for the dynamics and synthesis and experimentation, because pharmacokinetics, ADMET is also very important because there are so many molecules which are failed after entering into clinical trials and all. So you can avoid those things by doing all the predictions over here itself. And so this is one of the very good approach where you can begin with. So either you can generate a ligand-based model or you can generate a structure-based model, screen the molecules, identify the molecule which are having the right features, do the docking and predict the binding affinity. Then molecules with good binding affinity, you screen whether they filter the or whether they pass the ADMET, and then take those molecules and then do the molecular dynamics. So in the molecular dynamics, you will find whether the complex is stable or not, and you can calculate the energies of individual complex and all, right? So that is one approach, identifying a hit molecule or something, right? The second one is you can also use this pharmacophore modeling for predicting the pharmacokinetic parameters. So predicting the pharmacokinetic parameters means you can identify the molecule, whether it can have a good absorption, good distribution or metabolism or excretion, or it should be free from toxicity. You can also do that. For example, you can predict the metabolic profile also, right? If you look at the metabolic process, the major role is taken by cytochrome P450. And among them, if you see, most of the molecules, like they will be metabolized by this set, almost 90% will go by 3A4, 2E1, 2D6, 2C19, 2C9, and 1A2. So these are nothing but the isoenzymes of cytochrome P450, right? So, but when you want to use this pharmacophore modeling for predicting pharmacokinetic properties, let us say for absorption or metabolism, so what is needed is you need to have a data of molecules, right? You need to have a data of molecules, that is chemical structures with good absorption profile, with good distribution profile, with good metabolic profile, or which are free from side effects or toxicities. So you need to have that data. When you have the data, then you take those molecules and then you generate and again you give the absorption values. So you have to give some values, absorption, distribution, metabolism, excretion, anything. You give some values, and then again the same what we discussed, the molecule, these values also, ADMET values also, it should be in different ranges. When you have it in different ranges, so it will see what is the position of the feature in this molecule with respect to good absorption value. So what is the position of this feature in this molecule if it is having poor absorption profile, right? So you need to have different ADMET values for individual molecule. So which are good, which are moderately good, which are bad. So then it will identify where should be the position of individual feature. So when you want this, right? So for that, what is needed? As I said, you need to have molecules with good ADMET profile. This is required. So without this, you cannot do it. So now we have, there are so many, almost 3,000 or 4,000 drugs are there in the market, which are approved by FDA. So comparatively, they have safe profile. So you can pick those molecules for which you have experimental data, and then you can give it to the software and ask you to select molecules which are having, I mean, it will generate a model for which, if you want a good absorption profile, one model you can generate, good distribution profile, you can generate one model. Like that for everything. Good metabolism profile means this model. So free from side effects or toxicity, this model. So you can collect the literature, check the literature and collect those values, and then develop a model. And then, so whatever molecules you have, if you have an unknown molecule, you pass through that. So if it is matching the similar features, then your molecule also will have a good ADMET profile. So in that way also, this pharmacophore modeling will help you in designing. So in the laboratory, like once you are done, if you are doing a master's or PhD, so then you can generate a models for these, and you can tell the active compounds what you are getting. So you can screen those models and you can tell that. So this molecule, the molecules are the project outcome. So whatever active molecules you are showing, so you can also add to your data that this molecule has chances of having high absorption profile or high distribution profile and all. So that is an added data for your research, right? So in other way, you can also use it for target identification. So you have a molecule, but you don't know which target it binds. So again, the same way. So if you like, for example, you have done some antibacterial activity, but you don't know, you want to find this antibacterial activity is because of it is inhibiting which particular target. You want to know that. So then you can pick out some, by looking at the chemical features of a molecule, you can shortlist some 2 or 3 targets, and then you develop a pharmacophore model for those 2-3 targets, and you pass your molecules to them. So whichever is having similarity is there, more matches are there. So you can tell that. So this can be the molecular target. So because as it is inhibiting this target, so the mechanism might be, so it is this inhibitor, so and so inhibitor, you can just report it. So you can even cross verify the same with the molecular docking. So if the whatever molecule which is having similar features, so which are having required features, that if you do the docking, so then the chances of getting good docking scores will be higher. Right? So in that way, you can also use this for identifying the target. So of course, you can use for drug repurposing. So drug repurposing is also another huge research area. So where instead of identifying a new molecule and doing all the clinical studies, all the preclinical safety profile, instead of doing that, so whatever existing molecules are there, you can check whether it can inhibit, it will have the activity of that. So that is another new research area, repurposing or repositioning, right? Because the whatever drugs which are there, so in the market, so those were designed without understanding the structure of the active pocket, right? Of course, they're very selective to the target. I don't say that, or I cannot claim that these molecules are not right or something. Definitely, they are very good in activity. So that is the reason we all are surviving today. So, but they have a small side effects are there. Of course, still research is going on to minimize those side effects are also. Okay. So pharmacophore modeling can also be used to identify the target for your and de novo ligand design. So you can also, this pharmacophore modeling will also help you in designing a molecule, right? So once you have a pharmacophore model for the target, so you will know, like you need to know, like you will come to know, like you just see in the videos also. So some target is having, it requires 5 features or 6 features, something. So when you know those features, so along that you can construct a molecule with those features, and then you can check it. You can do the docking, and then you can do its synthesis and then check the activity. So de novo ligand design means, de novo means it is nothing but construction by step, right? Step by step, if a molecule, if a target says that it requires 2 aromatic rings, 2 hydrophobic group, 1 hydrogen bond donor, and 1 hydrogen bond acceptor. So we have a list of hydrogen bond donors, list of acceptors, list of hydrophobic groups. You can connect them by using some linkers or something. You can join them and see that whether the designed molecule is having the required features or something. So in that approach also, you can use this pharmacophore modeling for designing a new molecule, right? So there are multiple applications are there. So these are some of the applications. So in summary, we have seen it can be used for virtual screening. It can be used for predicting pharmacokinetic properties. So it can be used for target identification using pharmacophore approaches, de novo ligand design with pharmacophores. Okay. So these are some of the chemical databases which are available in public domain. So you can see there are millions of compounds are there, and there are crystal structures, 3D structures of proteins are also available. Millions are available in PDB. So what you just have to do, so you have to pick the right target, understand the target, chemical structure of the active pocket, what are the features, then you take those databases and screen them, and you can identify the molecule. And then those molecules, you make a series of it and then synthesize them in your laboratory, and then you can check the activity, right? So before going to that, of course, Lipinski rule of 5, I think most of you all are familiar. So if your whatever molecules you are filtering for synthesis, or you're going for the later stages, make sure that it clears the Lipinski rule of 5. So that will keep you in a safer side, like it will not fail in the later stages. Because so if it crosses this Lipinski rule of 5, so that means it will have a good ADMAT profile, and it will be free from all the side effects or toxicities. So you won't face much problems in the formulation or in the other phases. Okay. Yeah, so definitely we have discussed so many advantages and so many things about it. So does it have any limitations? Yes, definitely pharmacophore modeling has certain limitations, and it is very important to know the limitations so that it will help you to keep in your mind, so that you will take some necessary precautions while you are using it for your project. So definitely it has a successful and reliable applications are there, and there, like the drug molecules have been identified by using this pharmacophore approach. A systematic or straightforward way of constructing pharmacophore model is not available. So there is no one systematic approach. You have to follow this step-by-step process. So there is no systematic approach. A clear approach is not there, right? So different options are possible. So when I say that, if you take example, structure-based model generation, so what I say, a complex, you give the complex to the software. So in the complex, so when you talk about the active pocket, your active pocket may be surrounded, for example, maybe some 10 to 12 amino acids, right? So out of this 10 to 12, you cannot design a molecule which can interact with all the 10 to 12 amino acids. You may focus on 3 or 4 amino acids, or you will generate a model by seeing that these are the significant amino acids. So if your molecule is binding to this amino acid, the chances of activity will be high. But you cannot say that it is the right one. So it can be the next one also. Out of the 10, if you pick 3, so it can be other 7, or the amino acids may be in that 7 also, right? So there is possibility, and if you're trying to see another amino acid, the position of the feature will change, right? So that you need to do a lot of experimentation, and you have to see, you have to design a molecule which can interact with one particular amino acid, each and every amino acid in the active pocket, and then you have to do a lot of experiments, and you have to come out that if your molecule will interacting with this amino acid, so then only it can be a better molecule or something. So, but if you try to balance it here, then you have to look at other side, balancing the ADMET properties. Pharmacokinetic properties is also very important. So balancing this side and that side, so it may be again a questionable approach until unless you don't have a valid experimental data, right? So there is no accuracy in that part. Okay. Even if you look at the ligand-based models also, so you cannot consider, like for example, you are building a model with considering the like for example, you have a data set of activity range. That least molecule, the least activity molecule is 100 micromolar, and your highest activity is 1 micromolar. Just for example, you have built a model. So that means your activity range is 1 micromolar to 100 micromolar, right? So that means you don't know what will be the position of feature if the molecule is 1 nanomolar activity. 1 nanomolar means it is more potent, but you have taken 1 micromolar to 100 micromolar. So your model cannot predict what should be the feature, or it cannot pick the molecule which is 1 nanomolar activity. It may not pick because you have not taken. So something you can take it like a UV experiment, right? So if you want to know the unknown concentration, what you will do? First, you plot a calibration curve from the range you will select from which to which, right? So if you have generated a model, if you have plotted a graph for 10 to 50 micromolar. Now, if you have 100 micromolar concentration unknown, we cannot say how accurate it is. So it is also similar way. So it also has certain limitations. And another important limitation is conformation. So let us say when you give the software, it has generated 50 conformers only. So maybe 51st conformer might be the active conformer. So you have missed certain conformers for that molecule. So that is another limitation of it. So whatever conformer, and that conformation, if you remember the video, so after generating the conformer, you have to calculate the energy of that particular conformer. So whatever conformer your software is telling, if it is of higher energy, and if it is matching with the features, that is of no use again. So you should have the conformer which is low energy, and if it is matching the features, so then that will be of your advantage. So these are certain limitations what you have in the pharmacophore model. So it is always, of course, you need to do a lot of experiments to come up with a better pharmacophore model. So I'm not going in detail in advance. Okay. Before ending my talk, I just wanted to demonstrate one web server. It won't take much time. I think it takes hardly 5-10 minutes. Okay. So it is about PharmaGist. Okay. So it is a free server. Okay. It's not a software. It is a free web server where using this PharmaGist web server, you can generate a pharmacophore model. Okay. The approach what I'm using is ligand based. Okay. So what is required for you to generate a pharmacophore model using this PharmaGist web server is, so you need to have some 5-6 chemical structures. You need to have at least minimum 3 molecules. Okay. So whenever you're selecting, let us take the same example which we are familiar, COX-2, cyclooxygenase 2, right? So when you want to generate a pharmacophore model for COX-2 target, so what is required is you should always go for selecting the molecules which are in the market or in the clinical trial or in the preclinical trial, or if it is in the experimentation, you should pick the molecule which is very good activity, which is having very good activity. So you need to get the three-dimensional structures of that molecules. So you need to get the 3D structure of that molecule. So at least 3 molecules are required. Okay. Then, so those chemical structures should be saved in .mol2 format. So you have to save those structures in .mol2 format only, because the server can read only that .mol2 format. So you need to pick 3-4 molecules, minimum 3 molecules you should have, and then draw those structures, convert them into 3D, minimize them, and save them in mol2 format, and keep all those structures in one folder, right? So save all the structures. So what I did is I have collected all these molecules. Okay. So I have taken 6 molecules. I have given the name of the file name also. Celecoxib, heterocoxib, all these coxib, these are selective COX-2 inhibitors. Lumiracoxib, paricoxib, roficoxib, and valdecoxib. I have taken 6 molecules. These all are 3D structures I have taken. I have converted them into mol2 format. And then, so what you are supposed to do is you have to make them in a zip folder. Okay. So WinZip format is it has one software. It is a WinZip. You have to keep them in that. So you should not create any folders. Okay. All the 6 molecules you have to convert into a zip format. Don't put these molecules in one folder, and that folder you don't do it in a zip. Don't convert into a zip file. Directly you select the molecules and place it in a zip file. Okay. So once you place it in a zip file, so then what you're supposed to do is, so you have to go to this web server. I think I'll just open it directly. You can just type it in a Google Pharmagist. So it will directly comes. So Pharmagist web server. Yes, I'm not a robot. Yeah. So once you open this, it will come like this. So what you're supposed to do is, so you just have to load that zip file. Okay. Where it is. You can just keep it in one folder, right? Yeah. So this zip file, you have to load it. So once you load, so you can select how many output pharmacophores you want. So how many models you want it to like generate. You want to generate only 2 models, you want to generate 5, 10, 20, it is up to you, right? So then you give your mail ID. So you won't get your results immediately. So it will be sent to your mail. Okay. So I'm not submitting it. I'll just open the results which I got. Okay. I missed opening it before. Sorry for the inconvenience. Yeah. Yeah. So you will get a mail like this. So once you provide your mail ID, yeah. So you will get results like this. You have to click the link. Okay. Yeah. So once you click the link, so you will get like this. Okay. So what does it show? So you have a scores over here. So different scores are there. So I have given 6 molecules. Okay. So once you give the 6 molecule, so what it is telling is it has you have a different results. So this is the score when you align all the 6 molecules. That means here the good thing is you don't have to give the activity. Okay, so pharmacist, but provided you try to give your molecules which are in the market of clinical trials or something, so that will have. Yeah.
Excuse me, sir. Sorry to disturb you. We are not able to see you. I think the screen has freeze, sir.
Is it?
Yes, sir. We are only in that demonstration of pharmacist web server. We are not able to see. That's the reason.
Okay. So I think it is logged out. I don't know why it is.
Okay, we are in that the last slide which you had shown. After that, we were not able to see. In fact, someone has put it in the group also, in the chat box also.
Okay, I'll join it again. I'll join again. Yeah. Okay. 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 Yeah. Sorry for that technical inconvenience. So can anyone please tell me where I mean, you couldn't continue there, where you couldn't follow it up?
Sir, once you started with that online thing, that live thing, from
Yeah.
there, people could not see that.
Okay. Okay. So what is required is I have collected the 6 molecules. So these COX-2 inhibitors, I have converted to 3D structures and I have saved it in zip format. So once it is saved, so then you can just come to the Google. I'll just show you that is. Yeah. So you just Google in the type Google, you type it pharmagist. So you will get this pharmagist web server. Yes, I am not a robot. Please check the captcha. What is that? I think it is. Yeah, here it is open anyhow. So once you click that link, no, you will get this page like this. So here what you're supposed to do is you just have to upload that zip file, what you get. So all these 6 molecules. Once
Sir,
again,
your web, excuse me, sorry for the interruption. Sir, your web page is not been visible over here. You'll have to go to share screen and you'll have to check I mean, you'll have to share the other screen,
Okay.
like your web page.
Yes, yes, yes. Yeah. Okay. Sorry for that. Yes, yes. So once you,
It's visible now. You can
yeah,
go.
yes. So you just in the Google, you type pharmagist, right? So you will get this page. Okay. So once you get this page, you are supposed to upload the zip file what you have created. So this zip file, once again, I'm clearly stressing out, because if you just upload in a different way, it will not read and it will not accept it. So you need to have at least 3 molecules. All the 3 molecules should be in a 3D format. So you cannot upload 2D structures. You have to upload 3D structure. And those 3D structures should be in Mol2 format only. 3D structures in Mol2 format, and don't put it any folder. All these 5 or 6 molecules you convert into a zip format. You can download one software WinZip, and then so don't put these 6 molecules in a folder and then you convert into zip. Don't do that. So directly all these 6 molecules you select and put them in a zip file, and that zip file you have to load it here. So once you load it here, and then so number of output pharmacophore. So you can tell the server how many models you want. You want to generate 2 models, 5, 10, 20. You can generate up to 20 models. So then you give it to your mail ID. You have to give your mail ID. So once you give it your mail ID, so then it will send to your mail. Okay. Yeah. So you will get the results like this. So you have to click this link. So results, the results will be stored in web server itself. Okay. Yeah. So once you click the link, your results will be coming like this. Okay. So here, if you scroll it down, so you have different results are there. So I have selected 6 molecules. Okay. So this, it is giving, this is the first column is a score. Okay. And it is telling features, right? Now you can pick any model. So it is giving number of aligned molecules 6. So that means this model to generate this, it has given 3 models for this. So to generate these 3 models, it has taken all the 6 molecules. It has overlaid all the 6 molecules. It has seen what features are there in it. And it also has given individually. So etoric oxib, what features it has. So it has 9 features are there. That means 3 aromatic, 2 hydrophobic, 4 acceptor features are there. So like that individual molecule, how many features are there, it has given. And then it has overlaid on each other, all the 6 molecule, and it has seen what are the common features which are there in all the 6 molecule, and it has generated a model. So this model, this 3, it has given 3 models with the score 16.971 for 2 models, and the third one has 13.576. So if you feel that all these 6 molecules are potent and safe, and you want to pick you want whatever unknown molecule should have all these features, then you can select this. So what you are supposed to do, you have to click on this JMOL. If you want this pharmacophore model, just click on this. So then you will get like this. So it will show you that it tells that 3 features are very important. If you consider all the 6 molecule, what are those 3 features? So 1 aromatic ring should be there and 2 acceptors should be there. For every molecule, if it has to be a potent COX-2 inhibitor. So then you have to download this file. Okay. So download pharmacophore and alignment file. So if you feel that after doing all this study, if you feel that the score is less, or if you come across the literature search that one molecule, it is showing some side effects or toxicity, or you are not convinced with this molecule, and then you can go to this one, number of aligned molecule 5. So with these, if the server has considered these 5 molecule, so it has, the score is 25.456. So what are the different, I mean, it has done all the combinations and it has generated different pharmacophore models. If you feel that you can remove 2 more molecules, this is the scores, right? If you feel that 3 molecules are sufficient, so these are the pharmacophore models and these are the scores. So it has given you so many models with different combinations of drugs. So these are all drugs, okay? 6 drugs. So if you feel that this score is sufficient and If you all the features, all the 3 features which are required is pointed out by this server. If you want to take them, you can take that. So whichever feature you want, you can pick, or else if you are confused, you can pick first one from every. That means you can download this one. From 6, you can download the first one. For 5 aligned molecules, you can download this one. From 4 aligned this one. From 3 aligned this one. So if you don't do that, 6, 5, 4, that means you have 4 pharmacophore models, right? So once you have this pharmacophore model, now you have the model. You know what should be the position of individual feature in a molecule. So now what is the next task? Next task is you have to screen the databases and you have to find which are those molecules in the database has required features. Okay. So for that required features, you need to go for one more web server. So you have to remember these names. So first one is PharmaGist and this one is Zinc Farmer. Okay. Just Google it. So if you type the Zinc Farmer, you will get this. So you can click on this zincfarmer.csb.pit.edu. Okay. So once you come to the Zinc Farmer page, you click Zinc. So Zinc, it is a chemical database. Okay, Zinc database it is. So Zinc database, it has millions of compounds. So it has everything. That means it has compounds with reported values. It has compounds with even drugs, FDA approved drugs list it has. So it has millions of compounds with different experimental values. There are drugs with the compounds with experimental values. So there are also with vendors details, right? So once you identify the unknown compound, so you can even directly buy that from vendor, right? So it has so many applications. You can just explore the Zinc this web server, okay? And there are very good articles are there. You can just directly in Google Scholar, you can type Zinc and Zinc Farmer. You will get an articles on that in these both the websites, right? What all you can do, what all you can play from these web servers, okay? So once you click that Zinc, you will get this page. So here what you're supposed to do is, so you have to click this load features, okay? So click the load features because you have the features profile, right? Now once you have the features profile, that means what I have downloaded is this one. So I have downloaded 2, right? So with 6 features and with 3 features, okay? So I am taking the molecule, let us see the first one, which is 3 features I have downloaded. So you click the load features and you select this file, open, okay? So once you select that, so it will load and it will tell you. So these are the dimensions of your features. These XYZ dimensions of these features. As I said, position, right? Position of individual features are required. So it says that these are the features important. Okay. So these are the coordinates, XYZ coordinates of your pharmacophore model. And now once you load the features, after loading, you click the submit query. So once you click the submit query, so what does it do is, so it will screen the entire Zinc database. You can see how many molecules are there in this Zinc database. So it is going to compare 21,777,093. So that means that many millions of compounds are there in Zinc database. So it will screen the entire Zinc database and it will tell you out of these millions of compounds, how many molecules have the features which you require. Okay. So you can see the number. So it is telling that Zinc database has 5,651 molecule which have required features. So you can just click the save results. So it will download all that 5,651 compounds. So you will get download in a single file. So these molecules, you can take it for the docking. And then, so once you have the molecules which are having required features, now once you have, so these you go for the docking, and then you predict the binding affinity of these molecules. So these 5,000, once you predict the binding affinity, so you can cut down this 5,000 to maybe top 200 or top 300, you can take them. And then that top 200 or 300, you can do the ADMET screening, prediction of ADMET properties, and then you can come out with the molecules which are having very good ADMET pharmacokinetic parameters, so that you can take it for, you can identify the series of molecule. So which series you can synthesize it in your laboratory. So So from there you can pick based on your feasibility. So you can pick any one molecule and you can make a series of those molecules. You can just make some 10 or 20 compounds, and then you can check the activity of those compounds. So by this way, you can do the pharmacophore modeling by using these two servers. PharmaGist server will help you in generating a pharmacophore model, and then Zinc Farmer web server. So whatever pharmacophore model has come, that you load it to the Zinc Farmer web server, and then you get the molecules which are having similar features or the required features for this particular target. And then, so based on your requirement, you can continue with further pharmacophoric further computational calculations, and you can come up with the hit molecule. And you can go for the synthesis and you can screen them, and you can come up with some very good results. So coming to the end, conclusion. So pharmacophore modeling approaches, so they have been essential part of many drug discovery strategies. So though it has so many advantages and it has certain limitations also, but so there are certain drugs which are identified by using this standard protocol. So definitely it is a very good approach and very well validated and very much used method. So it has its own good strengths. So you can use it for multiple applications. As I said, you can use it for virtual screening, lead optimization, target identification, toxicity prediction, or you can create a new molecule and you can predict how well it can be. You can design a better molecule by following all these multiple approaches. Definitely, with one approach you cannot come up with a molecule. So you need to do you have to use different tools, and you can design a better molecule. So ultimately, so what I say is don't blindly follow the results what system is giving. So you have to put your logic and common sense. So system, like whatever data you will give the result. So here the main task is interpreting the results. So interpretation of results is very important. How well you are interpreting the results and how well you are getting the required data. So it always depends on you, right? Before closing, so the talk, I would like to thank for giving me this opportunity. So entire DSU team. So thank you very much, and I'll be happy to answer some questions.
Thank you very much, sir, for a wonderful session on pharmacophore modeling. The session is open to all the audiences, all the participants. If you have any doubt or any questions, kindly share it on the chat screen or you can ask. The session is open. I guess we don't have any question answer session right now or questions from the audience. So let me take this opportunities for thanking you once again, sir, for the wonderful session, for the audience and for the participants. We break now for the lunch. The lunch will exactly end at 2 o'clock. And by 2 o'clock sharp, we will meet again using the same link. Thank you. Ma'am, shall we start?
Yeah.
Welcome back, all the delegate speakers, to a wonderful session post-lunch. I guess you all are feeling fresh and rejuvenated to enter this post-lunch session. Let's right now start with the introduction on Speaker of the Day, number 3. The brief introduction will be given by Dr. Kalpana Devekar, ma'am, from COPS DSU. Ma'am, over to you.
Thank you, Anita. A hearty welcome to all of you again for the post-lunch sessions of the webinar. It's my privilege to introduce the first speaker of the afternoon session, Mrs. Srimati. She has completed her B.Pharm from PhD College of Pharmacy, Coimbatore, and M.Pharm from Madras Medical College, Chennai. She is pursuing her PhD from pharmaceutical chemistry at SRM College of Pharmacy, Kanchipuram. Her areas of research interest are computer-aided drug designing of steroidal sulfate inhibitors, molecular docking studies, molecular dynamics, and QSAR studies. Her technical skills include molecular dynamics simulations, pharmacophore modeling, and virtual screening and QSAR-ins. She has published 10 papers in various national and international journals and has presented posters in several national as well as international conferences. Mrs. Srimati has 7 years of teaching experience and at present she is working as assistant professor at SRM College of Pharmacy. Today she will be delivering a lecture on QSAR in drug design. Over to you, ma'am.
Thank you, ma'am. Whether the screen is visible?
Yes, ma'am, it is visible.
Okay, sir. Voice is also audible, sir?
Yes, ma'am.
Okay, sir. Good afternoon. So I'm Mrs. Srimati, working as assistant professor in SRM College of Pharmacy. So today I'll be talking about QSAR and its role in drug discovery. So in the first and second session, you would have listened to chemical substance or drug in the form of how a drug gets docked into a target, or how do you model a molecule, pharmacophore modeling. So now we'll be talking about quantitative structure activity relationship. So in this topic, you'll be viewing the drug in the form of a chemical and in the form of numbers. Okay, quantitatively we are going to see the structure. We are going to think of the structure in the form of numerical form. Okay, quantitatively, that is in numbers we'll be thinking it of. Okay, so you know that drugs are chemical substances. So either it might be an aspirin or propranolol or any kind of molecule. They are all chemicals, and these chemicals, they are made up of chemical ring, chain, and functional group. So these all linked together will give us the chemical structure. Okay, and drugs are three-dimensional. So this complementary shape or the three-dimensional structure of the molecule gives it a shape in which it is directed into the biological target where it gets docked. So this ring or the chain or the functional group which makes up a molecule targets a molecule into the target site. So it gives a shape or complementarity size to it, and based on the size and shape of the molecule, your molecule will go and land into the target. So when after our chemical substance has landed into our target, so it is like what is the body does to the drug. So after our drug has bound to the target, we get a lot of mechanical interaction, physicochemical interactions, and biological activity, pharmacological activity happening. So these are all result of after our drug have bound to the target. So how are we going to design our ligand or our chemical? Okay, so this is how you're going to view for the next 1 hour a drug in the form of a ring, a fragment, a functional group. So everything interlinked together and it makes up a molecule or a structure. Okay, so this is the definition. This definition I'm once again repeating, though you have listened to the first and the second sessions, for you to think hereafter the structure in the form of quantitation or in the form of numbers. So there are two major terms in medicinal chemistry. One is called as structural activity relationship. The other is called as quantitative structure activity relationship. So QSAR is a computational term and structure activity relationship is a pharmacological term, or how you can tell us how do you relate the structure to its pharmacological activity. So you would have studied about SAR of calcium channel blockers, SAR of AC inhibitors. You would have studied about structure activity relationship. So these structure activity relationship is derived for a series of drugs with the defined biological activity, and these series of drugs are going to be analogous to a series. That is, you have one structure. So based on that structure with differences in the functional group, substituting one functional group on one structure, what is the biological activity? You're changing it to another functional group, how it is going to be related to the biological activity. So relating between the structure and the pharmacological activity, and finding out what is the relationship between the chemical structure and its biological activity is called as structural activity relationship. Quantitative SAR or QSAR means quantitatively telling. So if you're having the structure, instead of performing the assay in the lab or finding out the log P value, the pi substituent value, or finding out the molecular weight or molecular mass. So after elucidation and then finding out these values, you know from the previous. For example, if you're having methanol, so what it is going to be its value. So you go to a higher series, ethanol, what is going to be its value? Okay, so quantitatively we can determine based on the previous information which is available. So that is called as quantitative structure activity relationship. Okay, so SAR studies relate the biological activity with the molecular structure of the drug. So after an SAR, by seeing a SAR, the chemist gets an idea to synthesize or to design a new molecule based on the previous information. So based on that, we design a molecule and then we take it for synthesis. So quantitatively, when you tell 50% of the validation, you're able to do it quantitatively. Then after that, so by having a validation, then you confirm on the molecule virtually and then you take it for synthesis. So that is the difference between the structure activity relationship and quantitatively estimating it. So here our relationship is going to be between a chemical structure and a pharmacological activity, and how we are going to convert the chemical structure into understandable numbers, and how we are going to identify the biological activity or relate these numbers with that of your IC50 value, which is going to be either in micromolar or in nanomolar. Okay, so how we are going to quantify, you will be converting the chemical structure in the form of number. Similarly, you will be getting biological activity, IC50 in the term of micromolar, nanomolar or picomolar. So these are also you're going to get numbers. How are you going to quantitatively estimate or obtain a interrelationship between these two, and what is the idea you're going to bring out of doing a QSAR study? So how does this concept of QSAR evolved, or from where, or who are the scientists who were involved in the evolution of QSAR? Okay, so you'll be knowing about Hanch and Fujita. So they worked on log P. Log P is a partition coefficient of a property. And what is the contribution of the substituent to each of the contributing to the partition coefficient of the molecule? And pi is the substituent effect of hydrophobicity. So these are very important terms in QSAR, like calculating the molecular volume, the surface area, then about the topological indices. So these are some of the descriptors. If you're already knowing QSAR, you'll be knowing about these terms. So just I'll give an overlay of how the history of QSAR and who are the scientists who were involved in making up of the QSAR equation. So this is a very few I'm limiting here, which is largely talked. And there are also other scientists who have worked in the similar field, who have bought all the individual fragments, giving it a quantitative value and summing up together. But I'm summing up the most important information I'm just giving. Okay. Next is Taft. Taft was a scientist who worked on electronic and steric parameters. Overton and Mayer, who worked on partition coefficient. How does partition coefficient relate to a drug absorption? Casamance, the central role of hydrophobicity in determining the protein structure. How does a protein structure get into a biological target? What are the factors for the structure to reach the chemical target? Craig, so this is called as Craig's plot. So what is the effect of substituent? If you're going to use, instead of chloro, if you're going to use a bromo substituent, whether it will be giving you a favorable interaction or a non-favorable interaction. So based on only the work on substituents. So this was done by Craig. Next is Topless. So the stepwise exploration of a series of physical chemical property, which is going to either increase the potency or decrease the potency of the compound. So that is called as Topless scheme. So these are some of the important parameters or important activities, like important functional groups, which are being quantitized and which are being used in the equation. Okay, so these are some of the scientists who have worked, but still there are many other works have also been done. These are very few. The important, like once you have done a QSAR equation, you will be going through the log P parameter, electronics, steric, hydrophobicity, and then what is the role of substituent. So these are the major parameters which are going to repetitively come when you're going to work with different structures for whatever target it might be. Okay, so this is the general idea of QSAR. So what is a chemical drug, and how you're going to see when you're going to do a QSAR, you're going to view the drug in the form of numbers, in which the system is going to understand. Okay, so that is called as, that is how you have to think when we are talking about quantitative structure activity relationship. And the outcome of performing a QSAR model is you're going to tell that the structure whichever you're going to design is going to have the pharmacological activity. So for that you will be having the previous literature sources. So in case of SAR, if you want to estimate the pharmacological activity of a homologous series, then you have to perform the experiment in the lab. Then you have to take the series of molecule and then you have to derive with the SAR. But with the available data, you can go for quantitative structure activity relationship, in which the same work which you have done in the SAR, you will be able to do in the QSAR. So this also reduces. So this is also like your molecular docking or molecular dynamics or pharmacophore modeling. QSAR also reduces your time, and also it helps in the better design of molecule. You have, when you're doing a QSAR equation, you would have done with some part of your validation of the particular molecule. So the rest is with the success of your QSAR model, you can go ahead with the synthesis in a you will be getting for biological activity. So that is the outcome of doing a QSAR. So how we are going to do a QSAR, or how it is being done normally. So a model development, so we'll be having the chemical structures. So this is our base. What we need to perform a QSAR model is, you need a series of structure, might be around 50 or 100 or to the minimum 30. Okay, so these many number of compounds with a defined biological activity. So these are the major criteria for us to perform a QSAR modeling. We need structures. The structures we need around 30 and above we need. If our data set is large, we get an accurate endpoint. And so we need the biochemical structures along with the estimated pharmacological activity. Okay, so the structure and experimental activity is the necessity for us to do a QSAR equation. And how do we take these structures? These structure, like whatever we are drawing in our 2D form, so these we convert into a term called as molecular descriptors. Descriptor is nothing but it is going to describe our molecule. For example, you draw a paracetamol structure, paracetoaminophenol. So the molecular descriptor will give you the information about the structure of paracetoaminophenol. So molecular descriptor is nothing but it is going to describe our molecule. So this is called as molecular descriptor. Why we need a molecular descriptor is, these are the numbers. We get the molecular descriptors in the form of numerical form. So these are the form in which the system is understanding our structure. So that is the reason we are converting our structure into molecular descriptor. Biological activity, you know, it is in the form of numbers only. Okay. So this is a necessity. So you get the chemical structure. After that, you calculate the molecular descriptors for the structure. We have online software such as ChemDes, or we can go with Paddle, which is used for developing the molecular descriptors. There you can develop from 0D, 1D, and then 2D and 3D structures. Okay, so converting the chemical structures into molecular descriptors. So after that, you can take that for developing a QSAR model, and you analyze it with the predicted activity, and finally you go for estimation or validation of the model that we have developed. So this is how QSAR works. So to do a QSAR equation, you need a chemical structure, you need a defined biological activity, we need the relationship between the structure and the biological activity, we need to convert the structure into descriptors, we need to then analyze it by performing a regression model, and then we have to validate it. So these are the major steps by which a QSAR modeling is performed. So to perform a QSAR model, it is already given with the OECD principle. OECD is an Organization for Economic Cooperation and Development. So they have given, they determine values for safety measure. It is an organization that works on safety parameter. So based on the OECD principle of QSAR, so there are certain 5 principles which are given, which should be followed while we are performing a QSAR model. So first is we need to have a defined endpoint. Second is the algorithm that we use should be an unambiguous algorithm. Third is we must be having a defined domain of applicability. Fourth is the validation parameter that we get should be of goodness of fit, it should be robust, and then it should be predictable. And principle 5 is if it is possible to mechanically interpret our model, then it is well and good. It is not necessary. The fifth principle is not necessary or not compulsory, but if it is possible, a mechanical interpretation of the model could be done. Okay, so these are the 5 principles on which our QSAR modeling lies on, and whichever model it is being done, it has to follow these 5 principles based on the OECD. So these are called as OECD principles for developing a QSAR model. So when we tell defined endpoint, endpoint is our pharmacological activity. So the pharmacological activity should not have any flaw in it. The data set that we are taking, the chemical structure and the pharmacological activity is called our endpoint. The endpoint must be clear. There should not be any flaw, or the value should not be it should not either be very different from that of the other value. The endpoint should be a defined endpoint because it is being done with different protocols and different experimental conditions. For example, if you're collecting a data set, we're going to collect it from different labs and from different experiments which are being done, but our target will be same. The structure that we are searching for will be the same. So when we are having the same structure and searching for the same pharmacological activity, it might have been done in different labs. So those lab, when you're taking those experiment endpoint for performing a QSAR model, the endpoint should be clear. So this is the first principle. Second is an unambiguous algorithm. Whatever algorithm we are going to use it to generate a QSAR modeling. So even if you are going to repeat it after performing the modeling, it should be having the same, showing the same result. That is, it should be able to reproduce. The model should be able to be reproduced. Third is, it should fall within the domain of applicability. Applicability domain means, for example, if you're having any one molecule which is already being defined and you're going to work on the similar series of structure. So when we are going to find out that whether it is going to fall within the criteria, we can cross check with the previous molecule. So if it is falling within the criteria, then we take those molecules as applicable. If it is not falling within the criteria, then we have to eliminate it. So we have to check, if you're going to work on the similar structure, we have to check that whether it will fall inside the applicability domain or it is going to be out of the domain. If it is going to be out of the domain, we should be eliminating those compounds and we should be taking only the compounds which are within the particular space. Next is goodness of fit, robustness, and predictivity. So this talks about the internal and external validation of our QSAR model. So once we have generated the model, we have to validate it both internally and we have to validate it externally. For example, if you're having 70 compounds, so these 70 compounds within where all the 70 molecules will be different structures. So these structures we have to first internally validate among our molecule, and then if possible we have to collect a similar data set and we have to do a external validation. So in the internal and external validation, if we do not have any flaw, it means that the model is robust and it will be capable. It will have the capability to predict further. So the fifth principle I've already told, it is a mechanistic interpretation. So if we need the mechanistic interpretation, we do it. Otherwise, if it is not possible for the model, it is not a compulsion. So this is how, based on these parameters, we have to perform our QSAR modeling, and these are the major principle or the main necessities of performing a QSAR modeling. So how do we go in a QSAR model? So first is we have to collect our data set. Our data set is going to be derived from 2 or 3 labs, and it is going to be derived from different molecules. So here the data set will be of might be of a homologous series of same molecule. So this data set on curation, when I tell curation, it means that it should be the structure, whatever we are going to take for the modeling, it should be accurate. Okay, there should not be any for example, it might be having the structure when you are taking into for a modeling purpose or for a molecular docking purpose, you're doing the protein preparation, you're doing the ligand preparation. So similarly, here in case of QSAR modeling, we have to check for tautomers, we have to check for isomers, and then we have to check whether the data, whatever we are going to give into the system, is going to be accurate. So there is also another term, GIGO, that is garbage in, garbage out. So whatever we give into the system, it is going to give us back. So we have to curate, or accurately we have to reduce the maximum flaws we can, and then we have to give our data set. So this is the first, and this is the this is the first step we have to follow while we are performing a QSAR modeling. So this is about our structure. So after we have identified the structure and we have curated the structure, we have to go for generation of descriptors. So this is a chemical structure. Now we are going to convert our chemical structure into a system readable format. That is, we are going to convert into chemical descriptors. So this descriptors, it could be of either one-dimensional, two-dimensional, or three-dimensional descriptors. Nowadays, there are also 4D, 5D, and 6D descriptors are also there. So normally we go for 2D and 3D QSAR we have done. So you can convert it into the structure, we can convert it into chemical descriptors. Then after converting into descriptors, we are going to split it into training set, test set, and then external validation set. So if you are having 70 compounds, we can keep one part of the component as a training, the other half as a test, and one portion we can give it for external validation. And then after splitting the data set into training, test, and external validation set, we apply the machine learning technique. We go for generation of the model. So we can use here statistical techniques like multiple linear regression, like partial least square. Then we can go for others like artificial neural network. We can go for support vector machine. So based on the data set that we are taking, we have to choose which type of machine learning technique we need and why we are going to apply that. We have to choose it. Then once we have arrived in the model, we have to do our internal and then external validation for the model. And we have to check whether it is falling within the applicability domain. And then we have to identify whether the model that we have done has the ability to predict the QSAR or to predict the efficacy of the structure for the pharmacological activity. So this is how normally we perform a QSAR. So first is we need a structure. We have to get an accurate structure. So the structure, we have to convert it into descriptors. And then we have to split the data set into training, test, and validation set. Then we have to apply the machine learning techniques. We have to go for statistical. So once we have arrived at the model, we have to validate it, whether it is having the internal and external validation. And then we have to check whether it is falling within the applicable limit. And if everything is being followed, then we can go for predicting the QSAR. So curation of the data set. So correctness of the input data, that is very essential by experimental scientists, because we are not going to have all the 70 molecules synthesized and screened in our lab. We are going to take it from different sources. So when we are going to take from different sources, we have to identify that whether the data set what we are taking is correct. So normally we get two types of errors. One type of error will be related to the chemical structure, and the other type of error will be related to the pharmacological activity. So either we have to check with the structure and we have to check with the pharmacological activity. So these are the commonest error which normally happen while we are performing a QSAR equation. We have to remove the inorganic organometallic counterion. So what type of errors we can face? And then we have to remove, we have to clean the structure, we have to check for ring aromatization, we have to check the structure as a whole for any errors it is having, and we have to also analyze with the endpoint, that is experimental activity. So if we get a correct data set, then we can feed it into the system, and then the other step will be validated. So this is the first step. So next is generation of descriptors. So the structures, once we feed into the system, it is going to be converted into descriptors. So these descriptors we have to generate. We can generate, as I told, 1D, 2D or 3D descriptors. So these descriptors are nothing but information about the molecule, describing the molecule. So this will be given in the form of numbers. They are quantitative description of the physical and chemical information of the molecule. So this is a descriptor. For example, physical and chemical characteristics, we get van der Waals volume, surfaces, lipophilicity, hydrogen bond related parameters. If we talk about the electronic configuration, we'll get partial charges, dipole moment, orbital energies. We'll get all this in numerical representation. We'll be able to generate topological indices. We'll be able to get the molecular fingerprint. We'll be able to get the fragment descriptors. So these are the different descriptors, and which descriptors we need for our equation, we have to identify. Based on our data set, we have to identify that which type of descriptors we need for modeling. So this is a simple structure and minimum number of descriptors. So this is with mole inspiration. Just if we feed the chemical structure into this particular software, it will be giving us different parameters. So here you can see the log P, TPSA, molecular weight, the number of violation, rotatable bond, then the volume of the compound. So you can see here each of it is given in number, in a numerical format. So this numbers will be taken. For example, the chemical structure versus log P. What is the relationship? Chemical structure versus the total surface area, chemical structure versus molecular weight. So if we have a similar series of this molecule together, around 50 or 60, if we have these values such as log P, TPSA, molecular weight, we can make a correlation between this particular structure and the pharmacology and the descriptor. So 1D descriptors will be giving information on the number of atoms, number of rings, number of carboxylic groups, number of rotatable bonds. If we go to 2-dimensional, we'll be getting information about the size, shape, and electronic distribution of the molecule. If we go to 3D, we'll get the 3-dimensional polar surface area, non-polar surface area. So based on the dimensions that we go, we get the information about the molecule. If we go for 4D, we'll get further information, 5D and 6D. So based on the dimension, we'll be getting the information of the molecule. So this is the second step. So first is we have to take an accurate data set. So second step is we have to go for generation of molecular descriptors. So this is how molecular descriptors are converted. In Google, if you type ChemDes, so here if you give your structure in the form of smiles, it will be giving us the descriptor value. Similarly, Paddle is also another software. So in Paddle, we can download and we can install it and keep. So in Paddle, if we give an entire data set, we'll be getting the descriptor value in an Excel format. So after completing these two steps, we go for splitting the data set into training and test. So in splitting the data set into training and test also, we have different types of splitting that we can follow. Either we can do a random splitting, it does randomly, it takes into training and test. So it is having an optimistic assessment of the model quality. The next thing is we can go with rational division of training and test. So here with some rationale, we go for division of the molecule into training and test. So based on that, we'll be we can arrive at a certain conclusion. So that is called as rational division. So next is k-means clustering. In case of k-means clustering, we cluster it in 2 groups and then we quantify it against another group. Then in case of sphere exclusion algorithm, we use either random forest or k nearest neighbor method, in which we use it with different, we exclude it as a sphere and then we analyze the other set of molecule. So like this, we have different type of methods to split it into a training and test method. We can use the machine learning. We can use these type of techniques to split it into a training and then a test. Finally, on modeling a molecule, we go to either linear regression, decision tree approaches, partial least square, support vector machine, artificial neural network, and then we can also go with random forest technique. So if we have a linear relationship between the structure and the pharmacological activity, we can go to linear method of QSAR analysis, like simple linear regression, multiple linear regression, principal component regression, partial least square. For example, if you're taking square of the activity, then we can go for non-linear methods. When we tell about non-linear method, we can go for support vector machine, support vector regression, PCA, k nearest neighbor, artificial neural network, random forest. So this is a non-linear method. So the method of the model development, we can decide based on our data set. We know of how the data set is related, the structure and the activity. Based on the relationship, we can choose either we have to go to a linear method of analyzing or a non-linear method of analyzing. So MLR is a commonly used method, multiple linear regression. In case of multiple linear regression, when we are having a chemical structure versus a certain biological activity, if we find there is an interrelationship between the structure and pharmacological activity in a linear way, we can draw a line and then we can find out the regression of this linear line, and this type of technique is called as multiple linear regression. So in the MLR analysis, the ratio of 5:1 is considered as absolute minimum. The number of independent variable is kept as low as possible, and the number of compound to be tested is also very low. So this is a commonest technique. We use genetic algorithm based MLR. So this is a non-linear technique. So either we can go with linear or non-linear type of model that we want to develop. We can go either with this statistical based, or we can go with techniques like support vector machine. So these type of regression technique we can use and we can develop a model. So once we have arrived at a model, then we have to perform the internal and external validation for the model. The internal validation is it is performed within the molecules that we are taking within our data set. We are going to check whether the molecule, whether our data set, whether it is fit for estimating the pharmacological activity. So in that case, what type of statistical parameters we normally use to identify whether it is a good QSAR model. So these are some of the terms you will have seen: R square, then we have Q square, adjusted R square, X squared, root mean square error, and bootstrapping or scrambling or Y randomization. So R square is like in the regression technique, we'll be getting the R square value of finding out the relationship between the structure and the pharmacological activity. In case we get a linear relationship, we'll be getting a R square value of above 0.6 or 0.7. So a model is told to be a perfect model when we get R square is equal to 1. So when R square is equal to 1, we tell that the model has a capability to fit the particular structure with its activity. But a model above 0.6 or 7 is considered to be a good model while we are performing a QSAR equation. it is not possible to obtain a model of R square is equal to 1. Even if we get 0.6 or 7 and above, we consider that, or we go on with rectification of the model to develop at least to bring it to around 0.67 or 8. So next is cross validation. So Q square is going to repeat the regression many times, and the R square, the difference between the regression R square and Q square cross validation, it should not be much. So for good predictability, R square minus Q square. So this difference should not exceed more than 0.3. It should be of a similar value. Next is leave one out. That is, each molecule is left out and R is computed to estimate the prediction. So in the internal validation, we leave one molecule out and it goes for prediction. And we can also leave many compound out and then we can do the prediction. So after doing leave one out or leave many out, so if our model is going to be predictable, so this type of statistical technique we do for internally validating our data set to estimate that whether our data set is having the biological, the relationship between the structure and the pharmacological activity. Next is we also do a Y scrambling technique. Y scrambling is shuffling the variable. We keep one as dependent, the other as independent, and we shuffle between the molecules and then find out what is the value we are getting in the Y scrambling value. So this is also repeated several number of times, and the value of Y scrambling that we get should be low compared to that of our R square and Q square value. So these are the minimum steps I can tell we do for internal validation of QSAR. In case of an external validation, from our data set itself, we have to split one part of a molecule into externally validation set, or we have to give a similar type of molecule as an external data set to externally validate it. So when we are giving an external compound into the data set, if our compounds have the ability to get validated externally also, then we tell that it has a capability both in the internal and external validation. We call that, so this model is going to be a better model. So we should not rely only on the internal validation. We have also to check out with some externally validated compound to increase the predictive power of our QSAR model. So here in this case, like R square predicted, if it is greater than 0.6, we call that this model is considered to be predictable. So this is about the external validation. And finally, when we are going to check it within a space, in a space, if we are putting the molecules and we have already developed a model, so if our model is going to fit into the domain, it means that it falls into the applicability domain and we can consider that particular molecule for estimated to be against with the biological activity. But if it is not lying within our applicability domain, then we tell that it is lying far from our domain. So this is considered. So this should be taken out from our biological activity. We tell that the structure is not having biological activity. So this is how we do a QSAR equation. Our data set is important. Whatever we are going to take as our data set should be curated or it should be accurate. Normally we face error with chemical structures. We face error with the experimental activity. So this part should be correct whenever we are going to do a QSAR modeling. So once our data set is correct, then we go for the structure. So we converting the structure in terms of molecular descriptors. So here we convert the chemical structure into descriptors. Next is, after taking our data set, we divide our data set into training set, test set, and then we divide it into validation set. So for dividing it into training test also, we have certain parameters such as random division, rational division, sphere exclusion we can use, k-means clustering we can use. So based on that, we can divide our data set into different. Then after that, we go for doing our modeling. So QSAR modeling, we use the statistical parameters such as multiple linear regression. Nowadays, we use artificial neural network. Then we use machine learning techniques, support vector machine. So based on these algorithm and based on whether we need a linear type of model or a non-linear type of model, we go for the modeling. We go on deciding for choosing which type of statistical technique we need to follow. Then after that, we internally and externally validate our compound. Internal validation is done with the parameters such as R square, Q square, then leave one out, leave many out, Y scrambling type of techniques. And external validation, we give an external data set and we estimate its predictability. And finally, we check it with the applicability domain. So this is how we normally perform a QSAR modeling. So how our model should be? So here the coefficient of determination R squared value is greater than 0.6. Then the cross validation Q square is greater than 0.5. R square predicted greater than 0.6. F value, the overall significance is better than 95. It should be better than 95%. This is called as Fisher value. Then standard deviation S should not be larger than compared to the standard deviation of the biological data. So this is how a QSAR, an acceptable model, should be having its value. So the different software that we use for QSAR is, in Schrodinger, we use Auto QSAR, field-based QSAR. Then we have BioPPSI open source software, Open 3D QSAR. Then we can use QSAR INS. We can use Build QSAR. So these are some of the software we use normally in the QSAR modeling. So how does the model come out, or output of a QSAR model? So here we can see the biological activity is equivalent to minus 3.15 plus the number of hydrogen bond acceptor minus number of hydrogen bond acceptor log P log S, molecular volume, molecular weight, stereo centers. So here we tell that if you are having a plus value, we tell that when we are going to increase the number of hydrogen bond acceptor in our structure, the biological activity is going to increase. The number of hydrogen bond donor we have to decrease so that the biological activity either will be maintained or it will not decrease. So this is how the positive and negative correlation to that of the biological activity is considered in case of a QSAR modeling. So this is how we will reach to a model, and this model this should be having a higher predictivity. The model should be in such a way it should be predictable. So here we are having training and test set. So we can see here the graph, a linear graph. We can see the molecules lying, training molecules and test molecule lying on the linear line, either up or down. It lies here, observed value versus calculated value. Then we can see here the validation parameter, the R square Y scrambling, Q square Y scrambling, model R square, model Q square. So this is also having R square and Q square, the blue color and the red color. It is being above, one above the other. So this is also being validated. And then here we can see the Q square LMO, leave many out. So this is also one method of internal validation of the compound. So this is how we'll be getting our figures in case of QSAR modeling. So with this graph, we'll be able to tell whether the model is able to predict or it has a prediction efficacy or not. So these are some of the references I've taken, from which we normally refer for any doubt we have. We refer for QSAR modeling, from which we learn a lot. So finally, I'd like to thank all the participants who had listened for the session. I'd like to thank our management, SRM Institute of Science and Technology, and I'd like to thank a college, SRM College of Pharmacy. I'd like to thank Dayanand Sagar University, Bangalore, for providing an opportunity to be as a speaker in this session. I'd like to thank the team of Dayanand Sagar University. Thank you so much.
Thank you, ma'am. Thank you so much for your subject details. Any questions? The question is open. The question session is open to the participants. Kindly feel free to ask any questions. You can put it in the chat box, or you can directly tell your questions. So let's move on to the next session, as we don't have much questions here. Yeah. I now request Dr. Rohidas to introduce the speaker number 4 of this session, who's the last speaker. Sir, over to you.
Thank you, madam. Good evening. I'm going to introduce a very renowned speaker, Dr. Jayaprakash Sangshetty, as a 4th speaker of our today's webinar series. Dr. Jayaprakash Sangshetty is working as an associate professor in YB Chavan College of Pharmacy, Aurangabad, Maharashtra. He has done his B.Pharm and M.Pharm from Aurangabad University and the Mumbai University. He was my immediate senior during my master's. And after that, he finished his doctoral studies from Baba Saheb Ambedkar Marathwada University, Aurangabad, under the able guidance of Dr. Devanand Shinde, who was the Vice Chancellor of Shivaji University, Kolhapur. So since last 18 years, with vast experience, Dr. Jayaprakash Sangshetty is doing excellent research in medicine chemistry. He's also an approved guide, and he's already having a lot of research students, almost 6 students in his laboratory for the PhD and several masters. And he has published over 200 international research papers, along with total cumulative factor of 320. So his H index is 31, which is a very growing very rapidly, and is doing excellent research in this molecular docking and computer aided design, as well as medicinal chemistry research. So he has authored also 3 books, and he has written 4 book chapters in different publications. He's also a reviewer of almost different international journals, more than almost 70 international journals. He's handling the revision activity, and almost 5 of his research papers were cited as the hottest 25 articles by the Science Direct. So he's a recipient of numerous awards, such as the Young Scientist Award by the Department of Science and Technology, Government of India. He has successfully completed various research projects, which are received from DST, UGC, and also AICTE. He has also received a Best Paper Award from the Elsewhere in 2015. And he got a Young Pharmacy Teacher Award from the Maharashtra State APTI in the year 2015. He also got this V-Life Best Publication Award in the year 2014, and also PD SETI Award for the Best Paper in 2009. And he has almost guided more than 30 M.Pharm students till now. So presently he's working as a board member for the studies of the pharmacy at Shivaji University, Kolhapur. And additionally, he's also handling international panel member for the project evaluation committee of the National Science Center, which is a Polish Ministry of Science and Higher Education, Poland, since 2015. And he has evaluated a number of projects of that government as well. So he's working as a brand ambassador for the Bentham Science Publisher since 2018. And his major area of the research includes drug design, synthesis of bioactive molecules, development and validation of the analytical methods for the drug and pharmaceutical dosage forms, application of the quality by design in the drug development, synthesis of polymers and their applications, along with the gene delivery. So he's one of my potential collaborator also during my research since last 7-8 years. And recently, I'm happy to say that recently has been included in the world's top 2% of the list of researchers, which was published by professors of the Stanford University. So that is another feather in his cap, and today he's going to speak about the antibacterial drug discovery, current trends, and future perspective. So let us welcome Dr. Jayaprakash Sansetti, and let us listen to his research work. So over to you, Dr. Jayaprakash Sansetti.
Thank you, Dr. Rohidas. Am I audible?
Oh, yes.
Okay. So, thank you, Dr. Rohidas, for the introduction. So, first of all, I'm thankful for the management for inviting me for this particular talk on antibacterial drug discovery. Now, before starting the session, the basic objective of this particular lecture is to overview what are the current trends going in this particular area, that is antibacterial drug discovery. So this particular presentation okay. So antibacterial drug discovery, the current trends and the future perspective. This is the topic which I'm going to talk about in this particular 1 hour lecture. Basically, this is a very vast area covering this particular area. Entire developments in this particular area within 1 hour, it is practically difficult. But in this 1 hour, I will try to cover the major advancements that took place in last almost 1 decade, and what are the future directions of the research in this particular area. Now, the intention of this particular lecture is basically to give the overview of the research which is going on in this particular area, since I'm working in this area since last almost 8 to 10 years, and different targets as far as antibacterial drug discovery is concerned. So for that, we must have the background of this particular subject. Then what are the established classes and what advancements have been taken place in last few years. Then what are the different targets that have been explored in last almost say 10 years, and what will be the future direction of this particular area, where how the future of this antibacterial drug discovery as far as the research is concerned. Now basically, if you see antibacterials, these are the most widely used class of drugs as far as the consumption of the drugs is concerned. And the need for antibacterial drugs, newer drugs, it is very much. As since again, some of the important aspects why there is a continuous need of newer antibacterial drugs, antibacterial agents. So some points I have mentioned. Now, as far as the drug discovery in this area is concerned, the drug discovery in this area is not much explored in last 15-20 years because of major issue of resistance of the drug. And to address that particular issue of resistance, number of changes, number of approaches have been followed. And these approaches, compared with other diseases, the success rate of antibacterial drug discovery, it is limited. And because of this, there is continuous need to explore the newer targets as far as antibacterial drug discovery is concerned. Now, I have given the number, how many drugs have been approved by FDA since last almost 1998. So the number is 25. Around 25 drugs have been approved by the FDA since 1998, which is very less number compared with other categories of the drugs. Now, if you see the era between 1970s to 1990s, it was continuous development in that particular field, and that development leads to number of new molecules in that area. But later on, that number of molecules entered, that is in between 1970s to 2010, the number has been reduced, and that particular reduced number, it is mainly because of the issue that I have mentioned, that is the resistance issue, which has not been fully addressed, and this issue has limited the development in this particular field. Now, what approaches people have tried, what are the disadvantages of this existing systems, and what people have made to improve that particular class. That particular discussion we are going through in this particular lecture. Now, as I mentioned, if you see the number of drugs which have been discovered in other categories, that is anti-cancer drugs, more than 650. As far as cardiovascular drugs, around 145, and in case of anti-HIV drugs, around 80. Now, if you see this number, the reason behind this less success of this particular antibacterial drug discovery drugs, antibacterial drugs, it is only because failures in the last stage or second, we can say in second and third phase of the clinical trial. And that particular failure is mainly because of the resistance issue that has been there with almost a majority of the drugs that has been discovered. Now, if you see this particular graph, so how this graph has been reduced with reference to the number of molecules which has been entered in the market. And again, I'm not showing this particular latter part, that is after 2012. Again, this particular graph is very small compared with this 2003 to 12. What it indicates, the number of molecules have been continuously reduced. And this shows that there is a tremendous need to explore the newer targets. There is a tremendous need to explore the newer approaches, either by combining the two fragments, either by combining the two subparts, or by discovering the drugs which acts by different mechanisms, that is dual acting antibacterials or multi-targeting antibacterials. And this particular need has always give rise to the novel areas of antibacterials, and these particular novel areas I am going to cover here. So if you see this particular list of drugs that has been discovered as far as launch dates are concerned, so very few molecules have been discovered in almost last say 1 or 2 decades. And again, this is the data which shows how many antibacterials have been entered up to 2011. And after 2011, some of the molecules I have highlighted, that is bezotoxumab, so it is approved in 2016. Then ceftazidime, avibactam, it is approved in February 2015. And tedozolide, it is an advanced linezolide drug, category drug. It has been approved in 2014. So very few molecules, they have been introduced in the market, and after introduction, they sustain in the market for longer time. So introduction, it is okay, but it has to sustain for longer time, and that long time usage must be proved, and that is not the case with the most of the antibacterials that has been discovered. Now, again, the different targets that has been explored as far as antibacterial drug discovery is concerned. So traditional antibacterials are different antibiotics. So cell wall inhibitors, beta-lactams are the categories. But again, a new class has been explored, that is inhibitors of peptidoglycan biosynthesis. So peptidoglycan, it is an integral part which is required for the cell wall biosynthesis of the bacteria. And this peptidoglycan, it involves synthesis involves number of biochemical steps and number of biochemical enzymes. Targeting the different enzymes in the synthesis of peptidoglycan, it has gained much attention in last 8 to 10 years, and few molecules have been entered in the clinical development, and these molecules are new hopes as far as antibacterial drug discovery is concerned. So most of the routes, most of the targets are already established. As far as protein synthesis is concerned, majority of the protein synthesis inhibitors are there in the market. Now, one more new approach through this protein synthesis inhibition, that is bacterial peptide deformylase inhibition. Again, fatty acid biosynthesis inhibition, it is the new era whereby most of the drugs, they have reached to the clinical developments. 1 or 2 molecules are in the further development, and there is again a hope from these particular categories. So if you see these all targets, so these targets which are already established, and some of the targets which has not been explored. And the targets which has not been explored, then knowing more about the targets, knowing more about the structure of the targets, designing the novel compounds from seeing towards the structure of the receptor or targets, and developing the new antibacterials, this should be the coming era of research as far as antibacterial drug discovery is concerned. Now, as far as updates of the established classes of antibiotics are concerned, what is the approach? Approach is existing drugs with certain modifications in the drugs with reference to the improved safety and spectrum of treatment, or the resistance, beta-lactam antibiotics, penicillin-like drugs, and After the 6 decades, beta-lactam is again a core pharmacophore that we are knowing. Majority of the drugs which are there in the market, still they are the advanced lactams, beta-lactams. Now again, a carbapenem, it is an analog which is a broad spectrum parenteral. Again, this particular carbapenem analog, it is an imidazole thiazole analog of this carbapenem, whereby it is having an activity superior to vancomycin and linezolide, and it is again in the developmental stage. Now again, a second carbapenem, which is again a modification of this particular original structure. Core is same with this particular thiazole nucleus. Now again, carbapenem C, which is an advanced carbapenem analog with this particular ethylene bridge, and this particular ethylene bridge is playing a significant role, whereby this ethylene bridge between the pyrrolidine nucleus and the isoxazole nucleus, it is incorporated to improve the stability towards the renal dehydropeptidase, which was a major issue as far as different carbapenems are concerned. Again, advanced cephalosporin with 18 to 16-fold more activity than the ciprofloxacin. This is again in phase 1 development. Now, as far as quinolone class are concerned, this particular quinolone class, this fluoroquinolone analog, which is again an equipotent activity as far as gemifloxacin is concerned. Then this is again a 4th generation broad spectrum quinolone analog, which has been developed specifically for the treatment of bacterial conjunctivitis. Now, some of the developments are in the category of tetracyclines, where there are some of the molecules which are developed from this tetracycline category, that is glycocycline, and this is active against gram-positive as well as gram-negative, and showing the activity against this MRSA. Then this is an aminoglycoside analog, higher analog, which is again aminomethacillin. So with spectrum of activity similar to the tigecycline. So these are some advancements, and whatever the structures I shown, these structures are again developed by doing some subtle manipulations or subtle improvements in the currently used antibacterials, and the logic for modification of structure was either to improve their spectrum of activity, or to reduce the toxicity, or to address the issue of resistance. Now, as I mentioned, different targets have been explored in the last 1 or 2 decades, and these particular targets we are going to cover, and these targets, how they are helping, and how these targets may be useful for giving some molecules in future. Now, if you see this Fab1 inhibition, as far as bacterial fatty acids synthesis is concerned, so fatty acids are playing a very important role in the different biochemical processes as far as any microorganism is concerned. Inhibition of this fatty acid involves number of steps, which includes fatty acid biosynthase enzyme, and this fatty acid biosynthase is of different types, that is ranging from 1, 2, 3, 4. And inhibition of this fatty acid biosynthase 1, it has gained an attention in last 10 years. And if you see these particular structures, these structures are proved as an Fab1 inhibitors, and this particular Fab1 inhibitors have been shown initial activities, and they are further in the clinical development. Now, both these molecules, they have shown the activities against this S. aureus. Then, if you see as far as pharmacodynamic profile of this particular drugs, they have shown some hopes for the further development as far as antibacterials are concerned. Now, another target, that is methionine tRNA synthetase inhibition. So this is an essential target that is mostly conserved in gram-positive bacteria. Output of the continuous efforts of medicinal chemists leads to a new inhibitor, which is again a quinolone analog with some modifications on the thiophene nucleus. And this particular molecule, it has shown some interesting results as far as different skin pathogens like S. aureus and S. pyogenes. Now, this is the most important target, that is cell wall biosynthesis inhibition. As I mentioned at the start, peptidoglycan, it is an essential bacterial cell wall polymer. That polymer is essential as far as maintaining the integrity of the bacterial cell wall is concerned. And if this particular polymer biosynthesis will get inhibited, automatically the bacterial cell wall integrity will be lost. And in synthesis of this peptidoglycan, it involves number of enzymes. It is a series of reactions. So more than 10 enzymes are required, more than 10 biochemical transformations are there in the synthesis of this peptidoglycan. All these enzymes are having their unique role. And inhibition of all these enzymes has gained much attention in last few years. And these enzymes are known as MER-A, that is muramidase A, muramidase B, up to muramidase Y. And these particular enzymes are the family of MER enzymes, which are known as MER ligase. Inhibition of any one of the enzyme, could be the attractive target as far as antibacterial drug discovery is concerned. Now, what people are doing nowadays, they are targeting one or two enzymes at a time. So the issue of resistance can be overcome. Multi-targeting has gained much attention in last few years. The reason behind this is the resistance issue that can be addressed to a greater level. Now, as far as this MER inhibition is concerned, either inhibition of a single enzyme or inhibition of 2 to 3 enzymes at a time, designing the different drugs and evaluating these particular different molecules for inhibition of single or multiple enzymes has gained much attention as far as antibacterial drug discovery is concerned. So these MER enzymes, which are the integral components in the bacterial peptidoglycan biosynthesis, they have gained much attention. Some of the molecules which I am showing now, they are in the clinical development. Now again, some of them are inhibitors of MER-A, some of them are inhibitors of MER-B, some of them are inhibitors of MER-A, B, C, D, and F. Now, either they are single enzyme inhibitors or multi-enzyme inhibitors. So these are the structures that are in the clinical development. Now, as far as this work is concerned, we have also started to work on this particular enzyme, that is MER ligase inhibitors. And to start that particular area, we have written one review, and that particular review has been published in Current Pharmaceutical Design. And this particular review we have published in collaboration with Mark Moloney, who is an expert in the antibacterial drug discovery from the Oxford University. And once we started writing this particular review, we got many ideas, and we have now started to work actually on this MER ligase inhibition. And on one of my projects, which is there from the SERP, I'm working on the multiple inhibitors of MER enzymes. And initial results show that some of the molecules that we have developed in-house, they may be fruitful for the further development. Now, again, the next approach, that is antivirulence. What exactly virulence is? It is nothing but the ability of any microbe to infect the human host. And successful inhibition of the virulence in the bacteria, it is considered as an important aspect as far as elimination of the antibiotic resistance is concerned. So that inhibition of the virulence, for inhibition of virulence, different targets have been proposed. And among the different targets proposed, MAC-C, it is one of the targets, and that VAC-C, it is a glycosyl transferase that is essential for synthesis of a lipopolysaccharide, which is present in the gram-negative bacteria. And inhibition of this VAC-C, that is considered as an attractive target, and most of the synthetic inhibitors that are reported, and this is one of the most, we can say, successful candidate, and again, which is a pyrazole analog, and with some modification of the original structure. And if you see this particular structure, this particular pyrazole nucleus, it is a competitive reversible inhibition of enzyme VAC. And this particular enzyme inhibitor, it may be having some scope as far as further development is concerned. And recently, while going through the news about this particular structure, I came to know that it has been entered in phase 1 clinical trial. Now, next target, which has been explored in last decade, it is bacterial peptide deformylase inhibition. Now, as we know, there are a number of antibacterial agents which are having the mechanism of protein synthesis inhibition in case of bacteria. Now, protein synthesis inhibition through a different mechanism, that is inhibition of the enzyme which is known as peptide deformylase. What this enzyme does actually, this enzyme catalyzes the removal of N-formyl group from the N-terminal methionine following translation during the protein synthesis. And this particular inhibition of enzyme, that is peptide deformylase enzyme, it leads to this particular process, which is an essential step in case of both gram-positive and gram-negative. And considering the significance, this particular enzyme in 2014, again, we started with the searching for the different targets. And in 2014, we have written one review on this particular target. That review has been published again in the current medicinal chemistry. Later on, while writing the review, we got some ideas and significance of this particular enzyme for the further development. And we have written many, we can say, we have developed some of the important molecules, and that particular work has been published in very good journals. Now, as far as this bacterial peptide deformylase inhibitors are concerned, these are some of the candidates which are in the clinical development. Glaxo is also working on this particular candidate, that is GSK 1322. If you see the structures of this particular inhibitors, so these are the structures which are in clinical development. And seeing towards the potential of this target, seeing towards the potential of these targets and the existing clinically we can say developed molecules, we have also worked on this particular target. And while working on the target, biaryl heterocycles we have considered as an important core, and on that core we worked and we got this particular result, and we have developed this some of the molecules, and these particular molecules again have proved fruitful. Now, further, this particular molecules needs to be explored with the available infrastructure, with the available setup. So we have given some primary clues about the potential of these particular molecules, and these molecules and the PDF inhibitory activity of this particular molecules we have published in the reputed journal in the medicinal chemistry field. So this is again another class, again which is based on the common core that is known as biaryl heterocycles, and this also we have published in a very good journal. So these are some of the publications which are based on the bacterial peptide deformylase inhibition. Now this is one target which we have explored first time in the Indian territory. So as far as peptide deformylase is concerned, so this is the target which is less explored. Some of the molecules are in the clinical development, and based on that hint, we started to work on this particular molecule. And with the help of one of the Department of Biotechnology in Pune University, so we have synthesized some of the molecules, and we have tested them for the peptide deformylase inhibitory activity. And this is the output, and 3 to 4 good publications from this, and these particular molecules may be further explored by some of the other research groups for the further development. Again, again on the same enzyme, that is peptide deformylase. This is the publication again, which is from CBDD, that is chemical biology design, from regulated science. Now, next area, next target as far as antibacterial drug discovery is concerned, that is known as quorum sensing biosynthesis inhibition. Now, what exactly the quorum sensing? Word quorum, we all are aware about the word quorum. So, what exactly happens if there will be a bacterial infection? So, bacteria through the signaling meshes call other bacteria to gather at one point, and this leads to increase in the concentration of the bacteria at a particular place. So, for creating a quorum at a particular place, there is one sensing mechanism, and this sensing mechanism, it is through a series of biochemical reactions. So quorum sensing, it is based on the interaction of low molecular weight signal molecules called as autoinducers, and this particular autoinducers, they give rise to a series of biochemical reactions. And inhibition of this particular quorum sensing, it leads to avoiding the quorum of bacteria at that particular place, and this will leads to antibacterial effect that has been proved. Now, there are a number of targets as far as this mechanism is concerned. And this particular target again has given some hints as far as the further developments are concerned. So biofilm inhibition is one of the mechanisms by which we can prevent this quorum sensing. Biofilm inhibition, we can prevent the bacteria as well as in the fungi also. Now, based on this target, again, some of the molecules which are reported with this quorum sensing inhibitory properties, which are again in the clinical development, based on some of the hints which we got from literature, we have worked on this target also. So again, linezolide molecule we have taken as a core, and we have prepared the ship base of this linezolide, and we have tested them for the biofilm inhibition of this particular synthesized molecules. We again, as I mentioned, biofilm inhibition, biofilm formation is one of the important aspects as far as quorum sensing is concerned, and this is the output of our research that we carried out. Now, as we have seen, these are some of the targets. Number of targets have been explored, but the targets which have proved fruitful in last 1 decade that I have highlighted till now. Now, what is the next approach? Besides targeting a single enzyme or single receptor, another approach which is known as dual action targeting, or we can say dual targeting concept. What is the objective of this particular dual targeting concept? It is just to address a common problem as far as antibacterial drug discovery is concerned, that is resistance. And to address the resistance issue, 2 molecules or 2 cores will be combined. These cores may remain intact in the body, or these cores may get separated once they enter in the body. And based on the type of mechanism, this dual targeting concept has been worked. So to show the exact mechanism, dual acting drugs. So A, it is linked with B with non-cleavable linker. So there is no biotransformation. A part, it will be bind to the target 1, and again B part will bind to the target 2. Now, both these targets, they are having an antibacterial potential. So inhibition or targeting both this 1 and 2, it will lead to the antibacterial activity. So if suppose the resistance has been generated due to inhibition of any one of the targets, then the second target will play their role of antibacterials. With that thought, at least the time duration of that particular drug activity can be enhanced, and the resistance time we can reduce. That is the idea behind this dual action. Now, as far as the dual action products are concerned, there will be cleavage either with the help of enzymes or without any non-enzymatic biotransformation or enzymatic biotransformation. A and B will get separated. A will get attached to the target 1 and B will get attached to the target 2. And with this particular basic concepts, number of drugs with a different mechanism of action, they have been cleaved or they have been fused with each other. And then these molecules have shown some good results, and they are in the clinical development. Now, as far as advantages of this particular approach is concerned, that is known as dual action or hybrid approach, we can call it as. Expanded spectrum of activity. If by targeting one of the important target, we may get the narrow spectrum activity, but with this hybrid approach, we may get the broad spectrum activity. We will get the synergistic effect, and we can either completely or partially address the issue of resistance with this approach, and it has been proved, and some of the molecules are in the clinical development. Now I will show some of the examples. There are many examples available in the literature, but some of the key important examples. Now there are some important aspects as far as how we can select the course and how we can go for this particular design. Okay, careful consideration of mechanism of action is needed. So these 2 cores which we are selecting, they should not antagonize the effect of each other. That is the criteria, and the synergistic effect is required. So consideration of mechanism of action, they should not antagonize their effect, and they must have a synergetic effect. These are the 3 important aspects while designing these drugs through this particular approach. Again, the first effort which has been made by GSK for using this dual action antibacterial approach, it is combination of cifamandole, which is linked with another antiseptic. If you see the structure, again, cifamandole, it is a cephalosporin analog. this cephalosporin analog, and omidine, which is again a 2-mercaptopyridine N oxide. So this part is an omidine. It is having an antiseptic property, and this particular core is a cephalosporin. Now this compound 1, this is effort of GSK researchers. Again, it has been entered in first or second phase of clinical trial. Now this particular compound, it is having an antibacterial activity. Again, some of the beta-lactamase inhibitors, and because of limitations of this cephalosporins, if this particular core will not work, then the role of this particular core will come into existence. That is the simplest mechanism. So two cores, they are clubbing to each other, and once they enter in the body, if one of the core is not acting because of the resistance or any other issue, then the another core plays their role as an antibacterial. So this is the simplest, but again, there are many issues, and these issues will be handled in the future, hope so, and we may get some good results from these efforts. Again, this is the example. Again, if you see this particular structure, it is an fluoroquinolone analog, and again, this is cephalosporin analog. It is a combination of the two cores with a different mechanism of action. Now, rifamycin quinolone analogs, this fluoroquinolones, they are showing their activity through the topoisomerase inhibitions, and rifamycins, they are inhibitors of the RNA polymerase. Now, workers at the Kamba Pharmaceuticals, they have recently disclosed this particular combination, and again, it has been further developed, and it is in the development in the clinical stage. Now, the idea is, this is the combination that is hydrazone linkage. This hydrazone linkage, it will be broken down inside, and the two core gets separated, and they will do their effect. And some of the promising results shown in the early studies has shown the hopes in the development of a very good antibacterial in future. Again, these are some additional examples. Again, this is the example from the oxazolidinone categories with the quinolone heterodimers. If you see the mechanism, oxazolidinone, they are having the protein synthesis inhibition properties, and these quinolones, as I mentioned, they are the inhibitors of topoisomerase. These two cores with a different mechanism of action. They are linked with stable linkage, and there will, once they enters in the body, these are the examples. These are some of the examples. You see this particular core, it is again a linazolide-like structure, and this particular fluoroquinolone analog. So they act by different mechanisms, and they are showing their antibacterial activity. That is a synergistic effect, since they are acting by two different mechanisms. Again, workers at the Theravance, they have developed another combination, that is cephalosporin glycopeptide conjugate. Now, this cephalosporin glycopeptide conjugate. So if you see two cores, cephalosporin, different mechanism of action, this glycopeptides, they are having interaction with this particular D-alanine terminals. Now, these two cores with a different mechanism of action, they have shown the synergistic effect as far as antibacterials are concerned. So there are many examples I have quoted. Again, this is the example which is in phase 2 clinical trial, and hopefully it will be in for the market in future. Now again, DNA polymerase quinolone hybrids. So inhibitors of the DNA polymerase and quinolone hybrids. This is the important combination that has been developed. And the researchers at Microbiotech, they have covalently linked fluoroquinolone antibacterial agent with DNA gyrase and topoisomerase with a DNA polymerase 3 inhibitory fragments. You see the two cores, MBX 500 is the example of this dual action inhibitors. And this particular combination, it is proved 500. It is more active against compared with the individual activity. And this hybrid structure, it has shown some scope for the further development. Now, seeing towards all these approaches, seeing towards number of targets that have been explored. If you see the success rate of antibacterial drugs, the comparative success rate, it is, if you compare with the other drugs, comparatively the The drugs which are entering in the market, they are comparatively less. And if you see overall scenario of antibacterials, more targets needs to be explored. Exploring the novel targets, exploring the structures of the receptors, designing the new drugs based on the structures of the receptors, combining the mechanisms, that is multi-targeting approach or dual targeting approach. That may be a future as far as antibacterial drugs are concerned. To conclude this session, basically, if you see resistance, it is the major issue as far as antibacterial drug discovery is concerned. And increasing resistance and failure of the drugs to enter in the market as far as antibacterials in the last stage of clinical development, it is a major issue. And the major reason is resistance. And to address the issue of resistance, finding the new novel targets, exploring the novel targets, identifying the novel targets, or combining 2 to 3 targets, or designing the drugs which acts on multiple targets, that will be the possible future as far as antibacterial drug discovery is concerned. So here, there is a critical need for searching for a new targets for the drugs with a new mechanism of action. So we can go for, we can address the resistance issue which is associated with the current antibacterial drug discovery. Now, as far as the different content that I have taken on the different references, again, I'm thankful to Principal Sir for giving opportunity to interact with you on this particular topic. Since it is a very vast topic, interacting or compiling the entire topic in 1 hour or 45 minutes, it is practically difficult. But still, I have given you the overview of entire antibacterial drug discovery program and different targets. Again, I am thankful to my guide, respected guide, Dr. Devan Chindel sir, Dr. Abubakar sir, who is a in-charge principal, who permitted me to interact with you. And again, thankful to your management of this university. And I'm thankful to Dr. Rohidas Arode, who is my collaborator since last 10 years. Thank you.
Thank you, sir. Thank you, Jayaprakash Sirji, for your wonderful session on antibacterial drug discovery, current trends, and future perspectives. I now hold the session for the delegates for the question answer. The session is open for all the delegates as well as the participants for the question answers. And if you have any questions, kindly feel free to put it in the chat box or to put it directly. Seems we don't have any questions here. So I request I request all the participants to fill the feedback form, which will be shared into the, which is already now shared into the chat box. Kindly fill the feedback form and wait for your certificate. Please give us few days time to give your certificates. You will be receiving your e-certificates on your mail ID, which you have provided. So feel free to fill the feedbacks form and wait for the certificates which are due to come. I now request Mrs. Rekha to give the vote of thanks for the entire session. Rekha. Rekha, ma'am.
Yeah, thank you, Anita. It gives me a great honor and privilege to deliver the vote of thanks to all the dignitaries of one-day national virtual seminar on computer-aided drug designing, a milestone in drug discovery. I would like to thank our Honorable Vice Chancellor, Dr. K.N.B. Murthy, to accept our invitation in his busy schedule, and I also express my heartfelt thanks to Dean of School of Applied Sciences, Dr. Pushpa Sarkar ma'am. I would like to thank all the speakers of the day. The first speaker, Dr. Parashuraman P., Assistant Professor, Faculty of Pharmacy, Ramaya University, Bengaluru, for his illuminating speech on computational tools such as computer-aided drug design and molecular docking and its significance in drug discovery. The second speaker, Dr. Manikanta Murahari, Assistant Professor, Faculty of Pharmacy, Ramaya University, Bengaluru, for his valuable speech on selection of pharmacophore and its modeling in drug design. So the third speaker of the day, Mrs. Srimati R., Assistant Professor, SRM College of Pharmacy, Tamil Nadu, for her valuable speech on QSAR and its correlation with biological activity and current trends in QSAR and its methodology in drug discovery. The fourth speaker of the day, Dr. Jaiprakash N., Associate Professor, YB Chauhan College of Pharmacy, Aurangabad, for his valuable talk on current trends in drug discovery, especially the targets of antibacterial agent. I once again thank all the speakers to accept our invitation in their busy schedule. I thank the Dean, Management, staff of College of Pharmaceutical Sciences, Dayananda Sagar University, to give an opportunity to conduct such an inspiring webinar. I also thank all the participants who attended the session and made the session successful. Thank you all.
Thank you, Rekha, for your vote of thanks. I guess Mahesh sir is waiting for the session to be handed over to him. So Mahesh sir, over to you.
Thank you very much, Anita ma'am. So I would like to request all the participants to fill the feedback forms. You will be receiving your certificates in a week time. Thank you very much. We are winding up with the session now. Thank you.
Thank you, sir. Thank you, all the participants, all the delegates, all the staff, and Honorable Vice Chancellor, Dean ma'am, and our respected principal, sir. Thank you, one and all, once again, for the wonderful session. Thank you.